Okay, hi everyone. I see that we have 26 out of the 32 students who originally registered for this webinar who have now come into the private Zoom webinar room. Welcome guys. And thank you for being on time as always. So this is going to be the next in our series of advanced level biology revision webinars covering in this case the subject of the control of gene expression. Now, again, in keeping with the format of the previous webinars, those of you who are in attendance, can you please make sure that if you have any questions during this webinar, please don't type those into the Zoom webinar chat. Instead, if you could type those into the Q&A box in your Zoom webinar interface, that way I won't miss the questions that you ask as the chat box scrolls by, and I will address those questions at the end of this webinar. Now, again, this is a webinar which is designed to assist students with their revision for their exams who are studying under the AQA Advanced Level Biology specification. But again, as with the previous webinars in this sequence, the content of this webinar is also going to be equally applicable to those of you studying under OCR or EdXL or any other exam board for advanced level biology. So I would encourage you guys, as always, to have your specification document open either in electronic format on your desktop or in hard copy format, just so that you can follow along with the accessible learning outcomes from your spec. Now, in the AQA specification document, which you're seeing here, this topic is topic 3.8, which is an A2 or advanced level topic on the subject of the control of gene expression. So these are the topics that we're going to be going into roughly in this order. Now again, as with the previous webinars in this sequence, if for any reason we don't get to cover all of these ideas and processes and concepts by the end of the 3-hour limit for this webinar, I will leave it up to the attendees to decide whether or not they'd like me to continue for a further period of time to cover the outstanding topics, or if they'd prefer me to schedule an additional part 2 to this webinar that they can attend to cover those topics, or if they'd prefer for me to upload an additional video which I shoot offline for them to cover these topics. But again, we'll see what we can do during the course of this webinar. Now, again, there are a lot of things to cover in this webinar, so let's get started. But again, if you guys could please make sure that any questions go into the Q&A box and not in the chat box. Now, I am expecting some students from the LeSwap consortium of schools to attend, and again, as with the previous webinars in this sequence, by previous arrangement with them, they've agreed to have their webinar chat displayed up on the screen, but as yet none of them have yet logged in. So as and when those students log in, I will bring up their chat up on the screen for you. So let's get started then. Now, the first topic that we need to address in this webinar is the subject of gene and chromosomal mutations. So, what is a mutation then? Well, mutations are sudden, significant, random changes in the genetic constitution or DNA of a cell, and these introduce an unpredictable form of variation. Now, mutations occur in all groups of living things, and mutations may be restricted to either changes to a single gene or may affect a number of chromosomes within a cell, and we're going to be looking at examples of those. So here we can see one example of a rare mutation which results in a goat with 4 horns here. Now, an organism affected by a mutation, like this chap over here, is termed in biology a mutant. Now, mutations in an organism's non-reproductive cells, a somatic mutation in other words, these affect that one organism only and cannot be inherited. Now, in contrast to that, mutations in an organism's gametes or sex cells is referred to as a germline mutation, and of course these mutations can be inherited to future generations of that organism. So, whenever a new form of gene results from a mutation, there may be more rapid changes in a species, and of course, mutations account for much of the significant variation that is the basis for the speciation, which occurs in evolution by natural selection. So, when a cell's dividing during mitosis or meiosis, there is a possibility that changes can occur in the chromosomes now, or in the genes, individual genes. Now, these changes again are mutations, and these are fairly common, particularly when cells are undergoing mitosis or meiosis, and these can lead to the formation of new genes. Now, if the new genes confer some form of selective advantage to the organism, such as, for example, better hearing or better sight to detect predators, or better camouflage to avoid predators, for example, these will become part of the genetic makeup of that species, or at least are more likely to do so, because the individuals possessing those beneficial mutations are more likely to survive and reproduce and pass on those mutations to their offspring. So again, one of the examples that we've mentioned before in previous webinars of this process is the evolution of antibiotic-resistant bacteria. So mutations do occur naturally, and particularly during the process of cell division, but the causes of these mutations are such that these spontaneous mutations generally occur at relatively low rates in a population. So mutations are generally caused by what we refer to as mutagenic agents. Now mutagenic agents can be viruses, they can be various types of chemicals or radiation, but all of these different types of mutagenic agents can be responsible for mutations, either gene mutations or chromosomal mutations. Now, different types of radiation, whether we're talking about electromagnetic radiation of particularly high frequencies like gamma and ultraviolet, or if we're talking about nuclear radiation such as alpha particles and beta particles, these all have effects on living cells. And these effects, typically in the case of electromagnetic waves, will depend upon the wavelength and also the dose of the radiation received. Now, the reason for this is that the energy carried by an electromagnetic wave in general is proportional to its frequency and inversely proportional to its wavelength. And this is the reason why the particularly high frequency or short wavelengths forms of electromagnetic waves, such as gamma rays, are particularly damaging and particularly prone to cause mutation in cells. So the DNA, for example, of and the chloroplasts of spinach cells, for example, are particularly susceptible to certain forms of radiation, and these can be used to study the effects of radiation. But in fact, animal cells are even more susceptible to the effects of radiation. So we can define a mutation as a stable heritable change in the genetic material of an organism. Now, a gene or a point mutation can result from changes in the base sequence within a single gene, and these will give rise to new alleles of that gene. But deletions and substitutions within the base sequence of a gene, these are examples of gene mutations, or again, also known as point mutations. So genes code for proteins, as you know, hopefully, and these are synthesized at the ribosomes in the cell. And the synthesis of the proteins happens in accordance with the instructions in the nucleotide sequence of the gene. And again, the gene is simply a length of DNA which contains the specific sequence of nucleotide base pairs responsible for the coding or responsible for coding for a particular protein. So the deletion or the substitution of a base within the DNA code of a single gene, this will change the transcribed message in the messenger RNA, and these codons in the messenger RNA ultimately determine the nature and the sequence of the amino acids within the final primary structure or polypeptide chain of that protein. So the nature of the genetic information flowing from the DNA to the ribosomes for protein synthesis can be altered by mutation. So if we see here part of the DNA sense strand coding for a normal protein, under conditions where this part of the gene is transcribed, we would end up with an messenger RNA molecule that would look like this. And then, of course, you have the process of translation by which the sequence of codons in the RNA molecule is then changed or translated into a polypeptide sequence of amino acids. So the nature of the protein is determined by the exact sequence of the amino acids in the polypeptide chain, which we refer to as the primary structure of the protein. And if the genetic information flowing from the DNA to the ribosomes for protein synthesis is altered by mutation, then it follows that the primary structure of the protein is also likely to be altered. Now, the amino acid chain will then bend and fold into a functional tertiary shape by means of secondary structural motifs like the alpha helices and the beta sheets and beta barrels. So again, if the amino acid sequence is altered as the result of a mutation in the gene, then it follows that ultimately the tertiary structure, and if the protein is part of a quaternary complex, then the ultimate globular structure and quaternary structure of the protein can also be altered. Now, you've probably heard, and ad nauseum, the phrase in biology that structure relates to function, and this is true of proteins as well. Proteins are essentially the functional macromolecular structural subunits which make up the majority of the functional parts of cells. Now, as such, if you change the structure of a protein, then you're likely to change or abolish its function. So many proteins are in fact enzymes which control metabolic pathways in cells, which in turn will influence the phenotype of the organism. So gene mutations that affect the shape of the active site of an enzyme will result in a loss of that enzyme's activity. So for example, the mutations of the genes that code for an enzyme involved in a chemical pathway leading to the formation of the pigment melanin will result in the loss of this enzyme and therefore an inability of those cells to form that pigment. And this leads to the condition known as albinism. And we can see here a few examples of albino organisms that have undergone that mutation. So let's have a look at firstly gene mutations in a little bit more detail, and then we'll move on to looking at chromosomal mutations. Now, gene mutations are effectively small-scale mutations, even though their effects can be quite severe. So a point mutation is a change of a single nucleotide base pair in the DNA. So one nucleotide can be substituted with another which contains a different base. So this is referred to as a substitution mutation. So only one base triplet codon in this case would be affected. So for example, a guanine containing nucleotide can be replaced with, for example, an adenine containing one. Now there are three possible outcomes for the codon from a point mutation. So you can either have a silent mutation, you can have a non-sense mutation, or you can have a missense mutation. So the examples of conditions resulting from point mutations are some of the conditions that we've mentioned in previous webinars and that we're going to make reference to in this webinar and others as well. So these would be things like sickle cell anemia, cystic fibrosis, colour blindness, and various kinds of cancers which result from a base substitution in a tumour suppressor gene. Now we're going to look at the topic of tumour suppressor genes in a lot more detail later on in this webinar. So a base substitution in a DNA triplet may not alter the amino acid sequence of a protein because of the degeneracy of the protein code. Now we talked about the degeneracy of the protein code in a previous webinar and a couple of previous webinars in fact, one on the subject of biological molecules and another on the subject of genetics and biodiversity and variation in organisms. But essentially the degeneracy of the genetic code refers to the fact that there's more than one triplet codon which codes for most of the amino acids found in living organisms. So a substitution, for example, that changes the triplet UCC to UCA will still code for the amino acid serine. So this type of mutation would be referred to as a silent mutation because it does not affect the ultimate polypeptide structure of that protein. So substitution of one base pair for another within a gene sequence usually occurs as a result of an error during DNA replication. Now most base substitutions will result in a single amino acid being changed for another at a specific position within the final polypeptide chain. So for example, if a substitution in DNA alters the mRNA codon from CUU to CGU, then the amino acid leucine will be replaced by arginine in that primary structure of the polypeptide chain. So this kind of change can have a minor effect or can have serious consequences if it significantly affects the protein's shape. So base substitutions may have no effect on the structure of the final protein, and we will talk about that in a bit more detail later. So a well-known example of the effect of a base substitution within the human genome is the mutation which leads to the sickling of red blood cells as a result of a change in the structure of the hemoglobin molecule to form hemoglobin S, and this is what leads to the condition that we call sickle cell anemia. So again, here is part of the DNA sense strand which codes for normal hemoglobin. When this is transcribed, you will get an mRNA strand with this sequence, and the translation of this mRNA strand would lead, in this case of these three codons here that we see here, to these three amino acids in the polypeptide chain. So the normal hemoglobin molecule results in the formation of normally shaped red blood cells. However, if we then take that same codon and compare it to the same part of the DNA sense strand that codes for abnormal hemoglobin, we can see here that one of these bases has been changed, and in turn the transcription of that part of the DNA sense strand will lead to a change in the structure of the mRNA as well. and this in turn will, in this case, lead to the substitution of the glutamic acid in this position of the polypeptide chain for the amino acid valine. Now this single change of this single amino acid in the primary structure is responsible for a rather drastic change in the normal hemoglobin molecule's structure, and this in turn causes a drastic shape change in the red blood cells, which leads to this sickle-shaped red blood cell here, which is characteristic of the condition that we call sickle cell anemia. So substitution of the nucleotide with the base thymine for the nucleotide with the base adenine has altered the coding for a specific single amino acid within that polypeptide chain, and in turn that's changed or replaced the glutamic acid with a valine in two of hemoglobin's polypeptide chains. So the mutant hemoglobin, which is called hemoglobin S, this functions abnormally, and this is what causes the red blood cells to become sickle-shaped when they encounter low oxygen concentrations. So other small-scale mutations may either add or remove a base pair from a particular gene. Now, these kinds of changes would alter all of the base triplets downstream from that mutation. So these are the kinds of changes that result in what we refer to as a frame shift. So one of these types of gene mutations is what we call a deletion, which is when one nucleotide in the gene is lost altogether. And of course, because the nucleotides are read in triplets, this can alter the reading frame of the gene, and this is usually irreversible and is unlikely to be undone by an insertion. But we also have addition or insertion mutations, and this involves the insertion or adding of one nucleotide somewhere within that gene sequence. Now these are usually caused by transposable elements or errors made during the replication of repeating elements, in particular the repeating of adenine-thymine sequences. So insertions in the coding region of a gene, this can alter the splicing of the mRNA, and this is called a splice site mutation, or it can cause a shift in the reading frame, which is referred to sometimes as frame shift. Now both of these can significantly alter the gene product, because both of these types of mutations will alter all of the codons downstream from the position of that mutation. Now insertions can be reversed by the excision of that transposable element. Now the deletion of one or more bases within a gene alters the DNA message in such a way that the reading frame, as we said before, in the mRNA is incorrectly deciphered during translation. So deletions alter the base sequence within the reading frame, and again this is referred to as a frame shift mutation. So if however we deleted 3 bases, this does not alter the reading frame, but it does result in the loss of a single amino acid from the final polypeptide chain. And of course the reason for this is that the genetic code is constructed in triplets of base pairs. So if you remove a triplet, you are in effect removing the code which codes for a single amino acid in that chain. So an example of a condition where this is involved is a type of cancer called retinoblastoma, which is a very rare type of cancer of the retina, and this arises as a result of a mutation in the retinoblastoma gene. So the RB1 gene is found in chromosome 13, and the mutation is generally a deletion of the RB1 locus within that chromosome. Sorry, I had to mute my mic there because I have a little bit of a cough. Okay, so here again is part of the DNA sense strand which codes for the normal protein, and here is part of the DNA sense strand in which a particular nucleotide has been removed. Now let's look at the consequence of that. Everything downstream from that, in terms of the triplet reading frame, has been changed by this deletion. So we go from a messenger RNA strand in the normal individual, which looks like this, to a messenger RNA strand in the mutant individual, in which all of these are shifted by the removal of that single base. And this in turn is going to drastically alter the polypeptide chain from that point onwards. As we can see here, we've gone from arginine, glutamic acid, proline, to arginine, and then serine and leucine. And again, the polypeptides further downstream from that will also be likely to be changed as a result of this type of deletion mutation. So all the amino acids coded for beyond the point of the deletion can be different, and this is a frame shift mutation. So for this reason, these types of deletion mutations generally have a much greater effect on the phenotype of the organism, in other words, the structure of the proteins, and again, because the structure relates very closely to the function, this can often lead to the formation of completely non-functional proteins, and this can have a huge, drastic, and seriously deleterious effect on the health of that organism. So, to summarise, this is what's happened. Now, again, we said before here, if we, instead of removing a single base here, if we were to remove 3 bases here, then we lose 1 amino acid from the polypeptide chain. So, we've looked at gene mutations. Let's have a look now at chromosomal mutations. Now, chromosomal mutations, in contrast to gene mutations, are much larger scale in their effects because they involve whole chromosomes rather than just individual genes. And of course, a chromosome will contain many, many genes. So, in general, there are 3 types of mutation within a single chromosome. We can start by saying that there are deletions of large chromosome regions. Now, again, this is different from the deletion of a gene, of a nucleotide, or a series of nucleotides within one gene. This can involve the loss of many genes from a particular chromosome, and sometimes this can also lead to the loss of heterozygosity. There's also duplication, which can also be called amplification. Now, this can lead to multiple copies of all chromosome regions occurring within that same chromosome, and this increases the dosages of the genes located in them, and in turn can increase the amounts of the proteins coded for by those genes being transcribed and ultimately translated. There's also inversions, and these can reverse the orientation of a segment of the chromosome, and of course this can have the obvious large effects on the phenotypes of that organism. So there are 2 types of mutation which involve 2 non-homologous chromosomes. In terms of chromosomal mutations, there are insertions, which involve a transfer of a segment of one chromosome and its insertion into another non-homologous chromosome. There are also translocations, and these involve the interchange of chromosomal segments between non-homologous chromosomes. So, an example of this is the MYC proto-oncogene, which codes for the protein product that acts as a transcription factor and controls the expression of several genes involved in the cell cycle. Now, the MYC proto-oncogene is located on chromosome 8, and a particular mutation in this gene involves the movement of the gene to a different location. So this would be referred to as a translocation, and in this case, it's a translocation of that gene from chromosome 8 to chromosome 14. Now, this results in an overexpression of the MYC gene and a subsequent overstimulation of cell division. Now, as we'll see later on, the overstimulation of cell division due to a breakdown in the regulation of gene expression, particularly of oncogenes and proto-oncogenes, can result in cancer. And in this case, it does result in a particular kind of cancer. So the overexpression is implicated in this Burkitt's lymphoma, which is a particular form of cancer. So here is a diagram which illustrates what's going on there. So here we have the translocation of the MYC proto-oncogene from chromosome 8 onto chromosome 14. And so this results in chromosome 8 with a deletion and chromosome 14 with a translocated portion inserted into it from chromosome 8. So again, this leads to what we call Burkitt's lymphoma, and Burkitt's lymphoma is a form of cancer, like many forms of cancer, which results from the overstimulation of cell division, resulting in rapid and uncontrolled cell division. So let's take a look now at the next section of the specification. Now again, we do actually go into a lot more detail with lots more examples of all of these forms of cancer and various other forms of types of mutations, both gene and chromosomal mutations, in the relevant course modules on my website. Now I know that I think all of you, yes, all of you in attendance currently are registered for these course modules. So if those of you who want a bit more detail, or if you want to ask me some questions about this and explore this in a bit more detail with me, you can simply log in to this course module here, the control of gene expression, and you can then go into these individual lessons and access the resources there, the teaching videos and so on. And of course you can either post your questions as comments on those lesson pages. Alternatively, you can go into your private coaching area for this course module and you can request additional resources or ask your questions privately there. So let's go into the next section of the spec, which deals with, let's go back here. Okay, so we've just talked about mutations, chromosome and gene mutations. Now we need to start talking about the concept of cell potency and cell differentiation. So let's get into that. So animal and plant cells, hopefully as you know by now, arise as the result of sexual reproduction, and they begin life as a single fertilized egg, which is referred to as a zygote. Now in humans, the egg cell or the ovum and the zygote are both smaller than a full stop. So to become an adult organism, the zygote and the subsequent stages divide millions of times by mitosis, and we explored this in a previous or in fact we're going to explore this in a webinar on the subject of cells, where we get into the topic of mitosis in a bit more detail. Now in humans, the term embryo is used for the developmental stages up to the 8th week after fertilization. From that point, the embryo, the growing embryo, is now referred to as a fetus. So all the cells that originate from the zygote are genetically identical to each other. They have the same genome because they're produced by mitosis. Now what this means is that all of our body cells, or somatic cells, contain an identical set of genetic information in the chromosomes. So for example, nerve cells, kidney cells, bone cells, all the different kinds of cells that you find in the different kinds of tissue in the body, these all contain exactly the same genetic material, and yet they're all specialized, as we could see, to perform their specific functions. So what is it that causes this? Well, the reason why all of these cells are different, in other words, they are differentiated, is because each type of cell has a specific certain combination of genes which are expressed or switched on, and some others which are silenced or switched off. So for example, all human body cells have the genes which code for the production of insulin. However, in a nerve or a muscle cell, that gene is switched off because it's not relevant to the functioning of nerve or muscle cells. But in the cells of the pancreas, in the islets of Langerhans, that gene is switched on and expressed so that those cells then produce insulin. Now the process by which specialization of a cell or a cell type occurs is defined or called differentiation. Now you have to be quite careful here, because I see a lot of students dropping marks in exams because they're using the terms differentiation and specialization interchangeably. And like all scientific key terms, these have very separate and distinct and rigorous definitions which you need to be aware of. Now the way to remember it is that differentiation is the process by which specialization occurs, and specialization refers to the specific set of features that a particular cell type has, which enables that cell type to perform its specified functions within that tissue, for example. So most types of animal cells will differentiate at an early age, but many types of plant cells retain the ability to differentiate throughout the life of the plant, and this is one of the major differences between animals and plant cells. Incidentally, everyone, I must apologise if there is a bit of background noise on this in this particular webinar. Once again, it's a very, very hot and muggy day here in London, so unfortunately I've had to open the windows, and consequently there may be a little bit of background noise, so I apologise in advance if that gets a bit too distracting. If it does, please do let me know in the chat, and I'll see if I can do something about that during the next break. So, an organism's growth and development is genetically programmed, although it may be influenced by the environment. And so we go from a single cell by mitotic divisions to a bunch of a rapidly enlarging ball of an increasing number of undifferentiated, unspecialized cells. And eventually, these cells begin to specialize and differentiate into the cells of different tissues, which specialize and differentiate and multiply further. And it's at these phases that you start to see the development of the organism in its embryology. Now, embryology is an absolutely fascinating aspect of medical science, but it is incredibly complicated. Even organs such as the stomach, which doesn't strike most people as a particularly complex organ, those of you who go off to study medicine at university, you're going to see just how complicated the embryology and the development of the stomach is. And so embryology is very, very complex, but we've simplified it essentially in this diagram, which illustrates the kind of level of complexity and the general shape of the organism, the human, at these levels in terms of the number of cells involved. So what we're saying here is that throughout the life of the organism, special cells, which are referred to as stem cells and precursor cells, will undergo further mitotic divisions to replace worn out or damaged cells. And so we have differentiation in the embryological context here, but then in the adult organism you still retain some stem cells and precursor cells which are able to differentiate to form the specialized cells of specific tissues in order to replace cells which have reached the end of their life or have been lost due to damage. So let's have a look at this term cell potency now. So the term potency refers or derives from the Latin term potens, which means having power, and it refers to a cell's ability to differentiate into other cell types. Now, in general, the more cell types a cell can differentiate into, the greater its potency is said to be. So potency is also sometimes described as the gene activation potential within the cell, which, like a continuum, begins with totipotency, which designates the ability of a cell to develop into all types of somatic cells, as well as germ cells and also the placental tissues in the female, all the way down to unipotency, which is the ability of a undifferentiated cell to only differentiate and specialize into a single cell type. And you also have in between those stages the term pluripotency and multipotency, which are defined for you here. So I would encourage you guys to be very, very careful that you are familiar and that you recall the specific definitions of these terms, because as you'll see, they are mentioned in the exam board specification, not just for AQA, but also for the other exam boards as well. So to demonstrate when totipotency ends, we can split an early embryo into separate cells, and at stages up to the 8-cell stage, each cell can then go on to produce a complete embryo with all the fully differentiated tissues of the body. So therefore, at the 2, 4 and 8-cell stages, the embryo cells are still totipotent. Again, this means that they are able to divide and to produce all of the different kinds of stem cells and also the fully differentiated cells in an organism. So, for example, these three identical triplets originated at the 4-cell stage of the mitotic division of the early embryo. Now, experiments on animals have shown that totipotency in most animals is lost at the 16 or the 32 cell stage, and at this stage the cells are no longer totipotent, they are pluripotent. So this is by the process of what we call induction, which is the formation of specialized cells or tissues, and this is when the process of induction has begun in most animals. So the totipotent zygote and early embryonic stages can then develop into any of the 3 germ layers of the human. So in terms of embryological anatomy and general anatomy, we divide the layers of cells and tissues in a human into these 3 terms: the endoderm, the mesoderm and the ectoderm, and these are shown in this diagram here. So the totipotent cells of the morula will differentiate into cells, and again they are no longer totipotent, and these become either the blastocyst's inner cell mass, or ICM, or the outer trophoblast layer. So the trophoblasts are the cells that form the outer layers of the blastocyst, and these provide nutrients to the embryo, which then develops into a large part of the placenta. Now the inner cell mass is now pluripotent, and this is actually the source of the embryonic stem cells. So stem cells are undifferentiated cells which are found in multicellular organisms, and these can differentiate into specialized cells which can then divide through mitosis to produce more stem cells. So there's much research being done into the applications of stem cells because it has a huge potential for medical therapeutics. So there's a lot of research being done into exactly how these cells differentiate and under what conditions you can make them differentiate into the cells of specific tissues. And also the other aspect of this is, where do we find ethical sources for these stem cells? So stem cells, as I say, have the potential to treat a very wide range of medical conditions. So here are just some of the medical conditions which potentially could be treated by the use of stem cells. Now, stem cells are generally defined as either being embryonic or adult stem cells. So adult stem cells are found in various tissues. In adult organisms, the stem cells and progenitor cells basically act as a repair system for the body, which is responsible for replenishing the cells lost in the adult tissues due to either the death of those cells at the end of their normal life or due to damage to those tissues. Now, the stem cells taken from a fetus, including the amniotic fluid, are still classified as adult stem cells. Now, embryonic stem cells, or ESCs, these are isolated from the inner cell mass of the blastocysts, or the blastula, in non-mammals. So, in a developing embryo, the stem cells can differentiate into all of the specialized cells, the ectoderm, the endoderm, and the mesoderm, but they can also maintain the normal turnover of the regenerative organs such as blood, skin, or the intestinal tissues. So there are several accessible sources of adult stem cells in humans. You can go to the bone marrow, and this requires extraction by what we call harvesting, and this typically is quite invasive because it involves drilling into the bone, and typically the bone that's used is either the femur or the iliac crest. There's also adipose tissue, which contains the fatty cells, or lipid cells. Now, this requires extraction again by liposuction, but 1 gram of fat contains 500 times more stem cells than 1 gram of bone marrow. There's also blood, which is drawn from the donor, similar to a blood donation, and this is passed through a machine which extracts the stem cells and then returns other portions of the blood to the donor. And another potential source is the umbilical cord. Now, the youngest, most primitive MSCs can be taken from the umbilical cord just before birth. So there are many different applications of stem cells which potentially can be used for therapeutics in medicine. One such application is leukemia. Now, bone marrow transplant is actually a well-established use of adult stem cells, and these types of transplants are typically used to treat various forms of leukemia. And this diagram shows just some of the many cell types which are found in the blood cells produced by the hemopoietic system, which is composed of the hemopoietic stem cells found in the bone marrow. So we see here that there are many different kinds of cells that arise from these stem cells. Another potential source is the embryonic stem cells. Now, pluripotent embryonic stem cells again originate as the inner mass cells within the blastocyst. Now, the embryonic stem cells can become any tissue in the body, excluding a placenta. So only the cells prior to the onset of differentiation are totipotent, in other words, able to become all tissues and a placenta. Now, another potential treatment or therapeutic application of stem cells is in the treatment of paralysis. So you can see here functioning neurons which were developed from human embryonic stem cells. Now, these embryonic stem cells have been used in the search to treat paralysis caused by accidents. So these stem cells have been made to differentiate into functioning nerve cells which eventually can return some sensation and control to that person. So we take an embryonic stem cell and we make it differentiate into a neural stem cell, and we can then further differentiate that into different types of nerve cell. We can then add growth factors and nerve cells which replicate in vitro on a feeder layer of fibroblasts, and these nerve cells are genetically modified and tested, and then the nerve cells can be transplanted into the injured areas of the spinal cord. the transplanted nerve cells will then differentiate into motor and sensory neurons, and this can restore the normal sensation and also motor control in the body below that damaged area of the spine. So these types of treatments have been used in an experimental context, but they're still an area of major development in medicine. Now, obviously, one of the major sources of controversy over the use of stem cells is the ethics of these kinds of applications. So there are many moral and ethical dilemmas posed by this type of technology. So, for example, to obtain embryonic stem cells, the early embryo has to be destroyed, and of course this means destroying a potential human life. Now, the application of embryonic stem cell technology has such potential to alleviate suffering that this poses something of a dilemma, because it focuses the decision onto whether or not the embryo should have the full or the partial status and rights of a living person. Now, obviously, some people will object to the use of transspecies technologies, such as these shown in the photo, where we have here human embryonic stem cells which are being multiplied as a colony on a mouse embryonic fibroblast. So this is a transgenic technology here, and again this has caused some controversy. So again, totipotency is the potential of a single cell to divide and produce all of the different types of differentiated cells in an organism. Now, spores and zygotes are examples of totipotent cells. Now, in the spectrum of cell potency that we saw before, totipotency again represents the cell with the greatest differentiation potential, and again toti comes from the Latin totus, which means entirely, and potens comes from the Latin for having power. So totipotent means having the maximum differentiating power. Now, pluripotency again is the potential of a stem cell to differentiate into any of the three germ layers. Again, the endoderm, such as the interior stomach lining and the gastrointestinal tract, and also the lining of the lungs. The mesoderm, which is the tissue layer which muscle, bone, blood, and the urogenital system belong to, or the ectoderm, which epidermal tissues belong to, such as the skin, and also the nervous system. So, pluripotent stem cells are the starting point for the production of organ-specific stem cells, and these potentially again can aid in the repair and prevent injury and or degeneration. So again, pluri comes from the Latin plurimus, which means very many. So cells which have the potential to differentiate into multiple but limited cell types are referred to as multipotent cells. So examples here would be a blood stem cell or a hematocytoblast, here that you can see in this diagram. So this would be a blood stem cell, and these are found in the bone marrow and also in the spleen, and these can produce the different varieties of blood cells by successive differentiation and specialization, as shown in this diagram. So when the hematopoietic system of stem cells, when these types of stem cells divide, one daughter cell must remain a stem cell to enable continued formation of other blood cells. Now, multipotent cells are found in many but not all human cell types. So, for example, adipose tissues, cardiac tissue, bone tissue, and the mesenchymal stroma cells, or MSCs, which are found in the developing bud of the third molar wisdom teeth in children. These are all tissues and cell types in which you can find multipotent cells. Now, the third molars could be collected at the ages of between 8 and 10 years of age to provide a good non-invasive source of mesenchymal stromal cells, and these can be then later developed into osteoblasts to generate bone, chondrocytes to generate cartilage, or adipocytes to generate fat tissue. So multipotent cells are actually capable of conversion into pluripotent cells and thereby give rise to unrelated cell types. So fibroblasts, for example, can be converted into functional neurons, and human umbilical cord blood cells have been converted successfully into human neurons as well. So the concept of the terminal nature of cellular differentiation and the integrity of lineage commitment is now being challenged by current knowledge. So eventually all cells may prove to be effectively totipotent, and we may be able to induce any type of cell to form all kinds of tissues. And this is obviously a potential area of research that could be of huge value in the future. So, a unipotent cell is a cell that has the capacity to differentiate into only one cell type. Now, it's actually currently unclear if true unipotent stem cells actually exist. Now, hepatoblasts, which differentiate into the hepatocytes of the liver, or cholangiocytes, which are the epithelial cells of the bile duct, are bipotent. So a close synonym for unipotent cell is the precursor cell. So you may see those terms used interchangeably in some sources. So epidermal stem cells are the most common form of unipotent stem cells in the body. Now these stem cells are found in the lower part of the epidermis, which is referred to as the stratum basale. We're going to look into this a bit later in this presentation when we start to talk about regenerative medicine in terms of things like skin grafting, although we do go into that in a lot more depth in the course module on my website. So in the interest of time and covering what we need to cover in this 3 hours, we may just skate over that, but we will mention that a bit later on. Now, germline stem cells, these are the cells which are able to eventually develop into the gametes. Now, the primordial germ cells, or PGCs, these are unipotent because they can only develop into the primary oogonia and spermatogonia in the reproductive organs, or the gonads. So induced pluripotent stem cells, or iPS cells, or iPSCs, these are a type of pluripotent stem cell which are artificially induced from a non-pluripotent cell, for example, an adult somatic cell. Now, the somatic cell, in order to do this, is subjected to the forced expression of certain genes and transcription factors which play a key role in determining the precise state of the iPS cells. So the genes are added to the cell using a retrovirus as the vector part of the inducement, and again we'll look at some of these genetic technologies later on in this webinar as well. So these iPS cells actually exhibit rather similar traits to those of the embryonic stem cells, but they don't require the use of embryos. So chromatin is reorganized in an iPS cell and resembles what we find in an ESC. So it's less condensed and it's therefore more accessible to genetic engineering. So epigenetic factors are thought to be involved in the actual reprogramming of somatic cells in order to induce pluripotency. And again, we'll talk about epigenetics later on in this webinar. So iPS cells potentially could have the same therapeutic implications and applications as embryonic stem cells, but without the ethical considerations of using embryos as a source for those stem cells. These iPS cells could be used in tissue transplants to avoid tissue rejection, and could also replace some of the animal in vitro models which are currently used in medical research. Now, the first methods of producing iPS cells actually gave very low replication rates and also early aging or senescence of those cells, but the techniques have rapidly improved over time, and the iPS cells have been found to be potentially tumorigenic. Approval for many of the clinical stages of research hasn't yet been forthcoming in many countries because of this reason of the tumorigenicity of these iPS cells. However, relatively recently, in September of 2014, the first ever iPS cell-based transplant into a human was conducted in Japan. So here is essentially the scheme or the method by which we can induce pluripotent stem cells or iPS cells. So we start with isolating and culturing host cells, which are yellow here in this diagram of the culture. So an example of this would be the mouse embryonic fibroblasts or adult human dermal fibroblasts. We then introduce the 4 ESC specific genes, which are iPS factors, into the cells by using a retrovirus vector, and again we'll talk about some of these vector techniques later on. Now the red cells here indicate the cells expressing the exogenous genes. Next we culture those cells, the iPS cells, using the same method used for culturing the embryonic stem cells, and this involves using mitotically inactivated feeder cells, which are here in grey. And finally, we then select the iPS cells by using typical embryonic stem cell markers, and then we harvest the iPS cells. So stem cells or iPS cells can be seen in this micrograph here. So we notice here that the cells do not look like any particular specialized type of cell. They are still undifferentiated. They don't have the kind of morphological features that you would expect of any specific cell from any particular kind of tissue. So in 2007, researchers successfully transformed the human fibroblasts into pluripotent stem cells, and then in 2012, researchers showed that human iPSCs could be produced from exfoliated renal epithelial cells which were recovered from urine. So obviously this method of acquiring the donor cells is non-invasive, easy to undertake, and is free from the kind of ethical and moral implications of some other forms of acquisition of stem cells. Now, the induction time for this is only 2 weeks for the urinary cell culture and 3 to 4 weeks for the reprogramming of those cells. Now, in this case, the iPS cell yield was up to 4%, and they showed good differentiation potential. So, in September 2014, as we said before, a female patient in Japan actually received a transplant of a retinal pigmented epithelial sheet made from iPS cells. And this was actually the first ever iPS cell-based transplant which was successful into a human. So, the induced pluripotent cells have been made from adult human stomach cells, liver cells, skin cells, blood cells, prostate cells, and urinary tract cells. So, what are the applications of this, or what are the potential applications of this? Well, the concept of organ regeneration. Well, we know that the liver is one of these organs that can regenerate itself. Well, potentially we could get other organs to regenerate themselves after damage. And so, obviously, the application of iPS cells in this context is receiving a lot of attention. So the first liver transplant ever was done in 1963, and it would be a great step forward if we could generate a complete organ like a liver, or even just a single lobule of the liver, as it would avoid any concerns about rejection or the shortage of donors, because we wouldn't have to resort to transplants to replace part or a whole liver. However, organs are made of more than one kind of tissue. So, for example, the liver has a complex blood supply and several other types of cell involved other than just the hepatocytes. So, at the moment, organ regeneration from iPS cells, even though it is there as a potential application, it is still a distant goal for research. Now, in contrast to animals, in flowering plants, mitosis is much more localized than in animals, and it only occurs in the meristem tissue, which is found in specific regions of the plant. So, for example, in the growing tips of the shoots and also in the tips of the roots. So again, the word meristem originates from the Greek merizein, and it means to divide. So meristem is a region of undifferentiated pluripotent cells in plant tissues which through mitosis give rise to new specialized cells and more meristem cells. So plants grow in girth due to the activity of the lateral meristem tissue found throughout the length of the roots and the shoots in the cambium tissues. But again, as we said before, in contrast to human stem cells, plant tissues can retain the ability to differentiate and specialize at any point in the plant's life. So, I see that we're coming up to the end of the first hour of this webinar, everyone, so I will give you guys a 5-minute break. Now, the next portion of this webinar will deal with the regulation of transcription and translation, and we're going to approach this in 2 halves because in the specification for AQA, we begin with a general discussion of transcription and translation and the regulation of these processes, but then we go on to the epigenetic control of regulation of transcription and translation. So I will give you guys a 5-minute break, and then we will come back after the break and we will explore that in a bit more detail. So we need to, before we get into the regulation of transcription and translation, we need to quickly reiterate a couple of key ideas about the genetic code and protein synthesis. So the bases on a DNA strand are read in groups of 3, which are referred to as codons. And again, we covered the idea of DNA structure and also the genetic code in a previous webinar on the subject of genetics and inheritance, but we also referred to it in the previous webinar on the subject of biological molecules. So if you wish to get into that in a bit more detail, have a look at those webinars and also the course modules associated with those subjects on my website as well. So we have the basis on a single strand of DNA here, and as we said before, the code will organize these into codons or triplets. Now, each group of 3 bases will code for a particular amino acid, and this is referred to as the triplet code. Now, the 4 bases which are present in the nucleic acids can produce 64 different triplet codes. Now, this is way more than the 22 different amino acids found in natural proteins. So the codons are used to produce single-stranded mRNA by the process of transcription, and the mRNA then retains the original DNA code for that particular polypeptide sequence. At that point, the mRNA will then leave the nucleus and travel to the cytoplasm, where either in the endoplasmic reticulum or in the free ribosomes in the cytoplasm, the process of translation can then begin. So the nucleus contains the genetic code, and then the nuclear pores in the nuclear membrane allow the mRNA to leave the nucleus, because the ribosomes are actually present outside of the nucleus. So you find the ribosomes both on the endoplasmic reticulum and also continuous with the endoplasmic reticulum can be found continuous with the nuclear envelope, but you can also find ribosomes free floating in the cytoplasm. So in order to conserve resources, the cell can switch genes on and off, and in fact at any one time only about 5% of the cell's genes are being expressed. Now, in all eukaryotic gene expression, or rather in all eukaryotic cells, gene expression involves the flow of genetic information from its origin in the DNA, in the nucleus, to the ribosomes, where the polypeptides can be assembled by the process of translation. And this makes up essentially the central dogma of biology. So we go from DNA to mRNA, and this is the process of transcription, followed by the process of translation, by which the mRNA is turned into a functional protein. Now again, transcription occurs in the nucleus of the cell and involves the formation of messenger RNA, which carries the complementary message from the DNA to the ribosomes in the cytoplasm. And translation occurs at the ribosomes in the cytoplasm of the cell, and this involves the translation of the messenger RNA sequence or code into a specific sequence of amino acids in the primary structure of a protein. Now, RNA always differs structurally from DNA in three important respects. So firstly, the RNA molecule contains uracil in place of thymine. The RNA contains ribose in place of deoxyribose, and the RNA is usually single-stranded, whereas the DNA is a double-stranded helix. So, gene expression can be controlled at either the transcription stage or the translation stage. So these can be subdivided into the following four categories. So we can talk about transcriptional control, post-transcriptional control, translational control, and post-translational control. And in each one of these cases, these are the mechanism by which the control is exerted. So in the case of transcriptional control, the process of transcription is prevented, so the mRNA is not synthesized. In the case of post-transcriptional control, the mRNA is controlled or regulated after it's been synthesized. In the case of translational control, the process of translation is prevented, and in the case of post-translational control, the polypeptide is modified after it's been synthesized. Now, before the process of transcription begins, the gene has to be activated by specific molecules that move from the cytoplasm into the nucleus. Now, for the most part, these molecules are proteins which are referred to as transcriptional factors. Now, all of these will contain one or more DNA binding domains which can attach to specific sequences of DNA next to or adjacent to the genes that they regulate. So there are hundreds of different transcriptional factors, and they control gene expression by controlling the rate of transcription. So there are two general classes of transcriptional factors. There are activators and there are repressors, sometimes called inhibitors. So activators will speed up the rate of transcription because they facilitate the binding of the enzyme RNA polymerase at the start of the target gene, and therefore they activate transcription. So we see how this works in this diagram here. So the activators are needed for RNA polymerase to attach itself to the start of the target gene on the DNA chain. The RNA polymerase, again, is the enzyme which joins nucleotides together to form the mRNA strand during the process of transcription. Now, each of these transcriptional factors recognizes and binds with a specific nucleotide sequence in the DNA molecule located in the nucleus of the cell. Now, once it's bound there, it can then activate that specific region of the DNA to start the transcription process. Now, at this point, the messenger RNA is then synthesized by the enzyme, and the genetic code that it copied from the DNA is then translated into a polypeptide in the normal way by translation in the cytoplasm once that messenger RNA molecule leaves the nuclear pores, or leaves the nucleus through the nuclear pores. So repressors, which are again also known as inhibitors, these will do the opposite. They slow down the rate of transcription by binding themselves to the start of a target gene, sometimes called the operator gene, and thereby preventing RNA polymerase from binding, and in turn this will of course prevent transcription. So when the target gene is switched off, in other words it's not being expressed, the site on the transcriptional factor that binds to the specific nucleotide sequence in the DNA is being blocked by the inhibitor molecule. Now, the presence of the repressor molecule prevents the transcriptional factor from binding to the DNA. Now, this in turn will stop the process of transcription from occurring, and hence the polypeptide cannot be synthesized. So, here we can see a summary of those two different mechanisms. Now, I'm going to leave that up there for a second or two for the attendees, just so I can take a sip of my coffee here. Okay, so let's move on to the effect of estrogens on gene transcription. Now, this appears in the specification in a couple of different contexts. Now, we're going to explore the idea of genes and their involvement in cancer later on in this webinar, but you'll notice that the role of steroid hormones such as estrogens actually appear in this section of the specification before we get to the idea of genes and cancer. So we're going to be getting into cancer in a bit more depth, but let's just quickly have a look at the role of estrogens on gene transcription in general. Now, transcriptional factors are themselves regulated by signals produced by other molecules. Now, it's known that the hormones can activate, or various hormones can activate transcriptional factors and thereby stimulate or promote the process of transcription of particular genes. So, hormones, therefore, have the power to activate some genes. So, here is the structural formula for estradiol, which is a common type of estrogen hormone. Now, these estrogens are a family of steroid hormones which are well known for their role in the mammalian estrous cycle, but they're also involved in the process of gene expression throughout the body. So, there are a few variations in the structure of these estrogens which are actually found in both males and females, and these affect specific genes which in turn control cell growth by affecting the process of transcription. So estrogens, because they are steroid hormones, they are lipid soluble, and because of this they can diffuse easily by simple diffusion across the phospholipid bilayer of the plasma membrane of various cells. So once inside the cell's cytoplasm, the estrogen can combine with internal cytoplasmic estrogen receptors to form the estrogen receptor complex. Now it's this complex that translocates into the nucleus and then binds to the DNA to regulate the activity of different genes. So in this sense, it's a DNA binding transcription factor. Now, the formation of the estrogen receptor complex can occur as the shape of the receptor's active site and that of the estrogen molecule complement each other. And this, again, is rather like a lock and a key. So you may have come across, well, hopefully by now you will have come across the lock and key hypothesis before. So there are two functionally distinct estrogen receptors. So there is the estrogen receptor alpha and the estrogen receptor beta, which are usually abbreviated ER alpha and ER beta respectively. Now, to make matters even more complex, these can then interact with a variety of different cofactors and signaling proteins to modify the estrogen receptor action by either enhancing, in the case of coactivators, or inhibiting, in the case of co-repressors, the target gene transcription. So this is a space-filling, computer-generated model of the structure of the estrogen receptor alpha. You see here these alpha helices within its structure, which are bonded to each other by these protein segments, but you also see these other regions of the molecule as well. Now, the ER alpha complex, which is a transcriptional factor, can act as an activator which allows the RNA polymerase to function. So the gene ESR1, which codes for the ER alpha protein, is found on the 6th chromosome in humans. So when the estrogen combines with to form the estrogen receptor alpha complex, which again is a transcriptional factor, this can change the shape of the DNA binding site on the transcriptional factor and thereby activate that factor. Now, the activated transcriptional factor can then correctly employ its DNA binding domain to attach to a specific sequence of DNA adjacent to the gene that's to be regulated. So the estrogen receptor beta, again, this is the complex which is formed by this, is a transcriptional factor. This acts as a repressor, and this works by blocking the action of RNA polymerase. Now, the final outcome of the activation or the repression depends upon the target cell and the function of the specifically targeted gene. Now, in the case of ER beta, this acts as a dominant inhibitor of the ER alpha transcriptional activity in cells that express both of these receptors. So the gene ESR2, which codes for ER beta, is found on the 14th chromosome. So let's quickly take a kind of introductory look at the link between estrogens and cancer before we get into the section of this webinar that actually deals with genes and cancer in general. So here we see a cutaway diagram of the anatomy of the mammary gland. Here we see the lobules, which are responsible for the formation of the milk by the mammary gland. Here we can see the ducts leading from those lobules. And these are the locations where lobular cancer can be derived from the breast tissue itself. You can also get ductal cancers, and these are in fact the most common form of breast cancers, and these arise as a result of the uncontrolled division of the cells of the ducts of the breast tissue. Now, another form of cancer can be the fibroadenomas, and these are benign tumours which are rather common, and these are non-cancerous in the sense that they are non-malignant. Now, estrogen receptors are overexpressed in about 70% of breast cancer cases which are said to be ER positive, and it's been known for many years that estrogens can increase the risk of breast cancers. Now, in addition to breast cancer, estrogen and the estrogen receptors have also been implicated in various other kinds of cancers, such as ovarian cancers, colon cancers, prostate cancers, and also endometrial cancers, which affect the womb. Now, advanced colon cancer is actually associated with the loss of ER beta and the predominant which is the predominant estrogen receptor in colon tissue. So colon cancer can be treated with ER beta specific agonists. Now, endocrine therapy for breast cancer involves selective estrogen receptor modulators, or SERMs for short, and an example of this is tamoxifen, which is a drug which behaves as an estrogen receptor antagonist in breast tissue. Another example is anastrozole, which is an aromatase inhibitor. Now, aromatase is the enzyme which can convert testosterone into estradiol. So there are two mechanisms by which estrogens and their metabolic products can initiate tumour growth. The first of these is the binding of an estrogen to its estrogen receptor, which stimulates cell division and DNA replication in mammary cells. The second is estrogen metabolism, which can produce genotoxic and sometimes mutagenic products, such as the oxidation of estradiol or estrone can produce products which can react specifically with the purines in DNA, adenine and guanine. Now, genotoxic substances induce damage to the genetic material in cells by interacting with the DNA sequence and structure directly. Now, the result of both of these processes is the disruption of the cell cycle and programmed cell death, which is referred to as apoptosis, and also DNA repair. So this overall can increase the chances of tumour formation. Now, estrogen receptor alpha is associated with more differentiated tumours, where the role, if any, of estrogen receptor beta has yet to be identified. So the different versions of ESR1 genes have been identified with single nucleotide polymorphisms, and each produces a risk of developing breast cancers. Now, tamoxifen itself, which is shown by this space-filling model here and also this structural skeletal formula here, this can produce an increased risk of uterine endometrial cancers. Now, this is actually quite common to many of the chemotherapies which are used to treat various kinds of cancer. These chemicals themselves do carry some risk of inducing tumours. Now, raloxifene, which is another SERM, this has also been shown within the same high-risk group when compared to tamoxifen to cause less side effects and to be more effective at producing, or at preventing rather, non-invasive breast cancers. But the problem here is that raloxifene has been shown to be less effective at preventing invasive breast cancers than tamoxifen. So, like DNA, most biologically active RNAs, including mRNA and tRNA, do contain self-complementary sequences that allow parts of the RNA to fold and pair with themselves to form double-stranded segments of RNA, which can form double helices. Now, these double-stranded RNAs are highly structured and can catalyze specific biochemical reactions and also inhibit certain other biochemical reactions. So within RNA, base pairing occurs between cytosine and guanine, but in RNA, again, if you remember, thymine is replaced by uracil. So, unlike DNA, which normally consists of long double helices, double-stranded RNAs are collections of short helices which pack together into three-dimensional structures which resemble proteins, and it's this which gives them some of their potential catalytic activities. So, for example, ribosomes, which are in fact chemical complexes which can catalyze peptide bond formation, these actually have an active site which is composed entirely of double-stranded RNA. So, once mRNA strands have been synthesized, gene expression can still be prevented by breaking down the mRNA before its genetic code can be translated into a polypeptide. So this would be categorized as post-transcriptional control. Now, the interference of the mRNA is known as RNA interference, or RNAi. Now, RNAi is a biological process in which RNA molecules, which are typically short double-stranded RNAs, can inhibit the expression of specific genes. Now, often this is by causing the destruction of specific mRNA molecules. Now, RNA interference is typically caused by two distinct groups of short mRNA. Now, these are called small interfering RNAs, or siRNA, and microRNA, which is usually abbreviated miRNA. Now, for the remainder of what we're going to talk about here, we're going to deal specifically with siRNA because it was siRNA that was initially linked to RNA interference. Now, the discovery of RNA interference and siRNA in 1998 was led to the award of the Nobel Prize to Mello and Fire in 2006. Now, unlike normal RNA molecules, which are single-stranded, siRNA molecules are short double-stranded molecules of approximately 20 to 25 nucleotides in length. Now, these have the ability to interfere with the expression of specific genes, as we said before. So here we can see a section of double-stranded RNA, and we start with a dicer enzyme which cuts up the double-stranded RNA into small interfering siRNA strands, such as these. And then another enzyme can separate the two strands by combining just one of the strands of the siRNA. At this point, the single strand of the siRNA can pair with the complementary base on a strand of the messenger RNA. And at this point, the enzyme can then cut the mRNA into non-functional fragments, and it's in this way that the RNA interference is effected. So this means that the mRNA can no longer be translated into a functional polypeptide in the normal manner. So by doing this, the siRNA in fact inhibits the expression of specific genes, as the polypeptide or protein that it codes for can no longer be made during the process of normal translation. So siRNA affects translation through the process known as post-transcriptional RNA interference. Now, as a result, of research into this area, many diseases we can see are actually caused by genes, and it may soon be possible to use siRNA and miRNA to block those genes and thereby prevent these diseases. So it may soon be possible to use these to prevent diseases, and therefore in 2001 it was shown that synthetic siRNA could cause RNA interference. Now since then there's been a huge surge in interest in harnessing RNA interference for biomedical research and also for drug development. So let's have a look at the epigenetic regulation of transcription and translation. Now remember, genetics is the study of biological processes and phenotypes and their functional molecules encoded as heritable gene sequence information, which we refer to as the genome. Now in contrast to this, epigenetics is the study of stable cellular and physiological traits and their underlying mechanisms, which are heritable by daughter cells and which result from changes in gene expression which are not due to the change or rather are not due to alterations in the DNA nucleotide sequence. Now the sum of all of these epigenetic changes in a cell are referred to as the epigenome, in contrast to the genome. So for example, if we take identical monozygotic twins, these will have identical genomes, but one twin may become obese and develop a genetic disorder, insulin resistance and type 2 diabetes, whereas the other will not. Now this phenotypic difference in these twins cannot be explained by differences in their DNA sequence because they have the same DNA, but it might be explained by a difference in their environment, in this case obviously nutrition. Now, cellular mechanisms which respond to the environment but which do not alter the DNA sequence are commonly referred to as epigenetic. So these include nucleosome positioning, histone acetylation, histone methylation, DNA methylation, and non-coding RNA activity. So the field of epigenetics is rapidly expanding because these mechanisms are known to be integral in defining the precise changes during the normal development, as well as maintenance of the final state of cell differentiation in multicellular eukaryotes. So the disruption of the normal epigenetic mechanisms can lead to diseases such as cancer, Alzheimer's disease, and so on. And the understanding of these mechanisms may hold the key for future successful treatments for these conditions. Now, powerful new drugs have already been developed using our knowledge of epigenetics and the genetic code, how the genetic code undergoes transcription and translation, and the epigenetic control of these processes. So before we get into the epigenetic control of transcription and translation, we need to make reference to some of the previously explored ideas about the way that DNA is packaged. Now, DNA is packaged by wrapping it around histone proteins into a structure called the nucleosome, and this is what we show in this diagram here. We start with the double helical DNA molecule. Now, this is an incredibly long molecule which needs to somehow be packaged into a space fitting within the nucleus of a cell, and this is the way that it's done. It's wound around these packaging proteins which form these units called nucleosomes. So each nucleosome is actually composed, as we'll see, of separate subunits of proteins called histones, together with other structural features which we'll look at in a moment. Now, an understanding of epigenetics actually depends upon the knowledge of the spatial relationship between the DNA and the histone proteins found in these packaging units called nucleosomes. Now, a single nucleosome actually consists of a portion of double-stranded DNA which is wrapped around protein complexes called histone cores. Now, each histone core is composed of 8 subunits, 2 each of H2A, H2B, H3, and H4 histones. Now, this protein complex forms a cylindrical shape that the double-stranded DNA wraps around with approximately 147 base pairs per nucleosome. So here we can see the kind of diagrammatic representation of a nucleosome, but this is a computer-generated model of the structure of a nucleosome, which was generated from data from X-ray crystallography studies. So, the X-ray crystal structure of a nucleosome core particle of chromatin shows in atomic detail how the histone protein octamer is assembled and how the 146 base pairs of DNA are tightly organized into a superhelix around this. Now, the histone proteins are perfectly designed for their job, so much so that the histones are nearly identical in all non-bacterial organisms. Now, even slight modifications in these structures can be lethal to the organism. So, when DNA is to be transcribed, the nucleosomes can slide away from that region of the DNA, and this opens it up to the transcription machinery of the cell, which can then cause the transcription of that DNA section into a messenger RNA sequence. Now, chemical modification, for example by acetylation or by methylation to either the histone proteins or to the DNA itself, will signal whether or not a particular region of the genome should be open or closed to that transcription machinery. Now, only if this genetic regulation is inheritable by daughter cells and does not change the nucleotide sequence of the DNA is it referred to as epigenic or epigenetic regulation. So, nucleosome formation and integrity is dependent upon the presence and maintenance of positive charges of the histone 4 proteins and the negative charge on the surface of the histone 2A histone fold domains. So only the names and the tails of the upper 4 histone subunits are shown here in this diagram. In reality, the lower subunits have the same structure as the upper subunits. So, acetylation of the histone tails affects the integrity of the association between the histone domains, and this in turn leads to weaker binding of the nucleosomal components. Now, it's this weaker binding which makes the DNA more accessible and leads to more transcription factors being able to reach that DNA. So, in organic chemistry, the methyl group is an alkyl group derived from methane and contains one carbon atom bonded to 3 hydrogen atoms. And this is usually represented like this as a space-filling model and is usually denoted CH3. Apologies guys, this 3 should be a subscript and not a whole number here. So, many proteins will undergo methylation, in other words, the addition of a methyl group. Now, protein methylation has been most studied in the histones, which when methylated can act epigenetically to repress gene expression. So protein methylation is actually one of the post-translational modifications that proteins can undergo. So methylation of DNA, on the other hand, also occurs primarily by methylation of the organic base cytosine. So there are several sites on histones that can be methylated. So the methylation of proteins typically takes place on arginine or lysine amino acid residues in a protein chain. So here we can see that arginine can be methylated once or twice, and this is done under the action of enzymes called methyl transferases. So the methylation of the ionic form of arginine can occur once or twice because 2 terminal hydrogen atoms can be replaced by a methyl group. So this is the ionic form of arginine, and we can methylate at these 2 positions to form dimethyl arginine, and you can see that in these 2 space-filling models here. Now lysine can be methylated up to 3 times, and again this is done under the action of those enzymes referred to as methyl transferases. Now again, we can see this more clearly in the ionic form of lysine here, and we can see the 3 locations on this terminal group where you find hydrogens which can be replaced by the methyl transferase by methyl groups, as we can see here. Now in contrast, the methylation of DNA in vertebrates typically occurs at the cytosine phosphate guanine sites, or CPG sites. So this methylation results in the conversion of cytosine to 5-methyl cytosine. Now genes with high levels of 5-methyl cytosine in the promoter regions are always transcriptionally silent. In other words, they are not transcribed. Now the formation of MECPG is catalyzed by the enzyme DNA methyltransferase. Now human DNA has approximately 80 to 90% of its CPG sites methylated, but there are certain areas known as CPG islands which are guanine cytosine rich. So in other words, they have a high guanine and cytosine content, which is made up of approximately 65% cytosine and guanine residues. And in these, none are methylated. Now, there are associated with the promoters of 56% of mammalian genes these types of methylated residues, and including all the ubiquitously expressed genes. So 1 to 2% of the human genome is made up of these CpG clusters, and there's an inverse relationship between the CpG methylation and the transcriptional activity, in the sense that the more you find these CpG clusters, the less transcriptionally active those regions of the DNA become. So, methylation of DNA and histones causes the nucleosomes to pack more tightly together, and this in turn prevents transcription factors from binding to the DNA, and therefore these genes are not expressed. So we've looked at methylation. Let's take a look at acetylation now. So, in organic chemistry, an acetyl is a functional group with the chemical formula COCH3, and again apologies, this should be a subscript. So, among the proteins that are known to undergo acetylation are the chromatin proteins, in other words the histones, and also metabolic enzymes. Now, this indicates that acetylation has considerably more impact on both gene expression and also on metabolism. So in some bacteria, it's been calculated that 90% of the proteins involved in central metabolism are in fact acetylated. Now, acetylation is closely associated with increases in transcriptional activation, whereas deacetylation has been linked with transcriptional deactivation. So these reversible reactions typically occur as co-translational and post-translational modifications of proteins. So for example, histones and also the p53 protein. So histone acetylation typically causes the nucleosomes to pack more loosely, and in turn this means that the transcription factors are more able to bind to these genes and therefore promote a higher rate of transcription. So histone acetylation and deacetylation, these are the processes by which lysine residues within the N-terminal tail, which protrudes from the histone core of the nucleosome, can be acetylated and deacetylated as part of gene regulation mechanisms. So these reactions are typically catalyzed by enzymes with histone acetyltransferase or histone deacetylase activity, and these are typically abbreviated HAT and HDAC. So acetylation is the process where an acetyl functional group is transferred from one molecule, such as acetyl coenzyme A, to another molecule, such as these histone proteins. Deacetylation is the opposite, where an acetyl group is removed from a molecule. So the histone tails insert themselves into the minor groove of the DNA and then extend through the double helix, and this in turn leaves them open for modification involved in transcriptional activation. So the mechanism for acetylation and deacetylation takes place on the amino group of lysine amino acid residues. Now these residues are typically located on the tails of the histones, again, that make up the nucleosomes of packaged double-stranded DNA. Now, the acetylation has the effect of changing the overall charge of the histone tails from positive to neutral. So histone acetylation, deacetylation and methylation, these are all essential processes by which the lysine and the arginine residues within the N-terminal tails of the histone subunits can contribute to the regulation of gene transcription. So there are many different sites on the histones that can be activated or deactivated in these ways in different combinations. Now, the cellular differentiation that we mentioned before is an important example of an epigenetic change in multicellular eukaryotic organisms. So during morphogenesis, or the coming into being of form, totipotent cells or totipotent stem cells become the various pluripotent cell lines of the embryo, and these in turn become the differentiated cells such as neurons, muscle cells, epithelium and the endothelium of blood vessels, etc. So this cellular differentiation is achieved by the activation of some genes and the switching off or the inhibition of others. So DNA methylation is essential during embryonic development, and in somatic cells, patterns of DNA methylation are in general transmitted to daughter cells with a high degree of fidelity. So cellular differentiation is an epigenetic change because it modifies the activation of certain genes, but not the sequence of the DNA. Now, additionally, the chromatin proteins, the histones for example, associated with DNA, can be activated or silenced. So this is why the differentiated cells in a multicellular organism express only the genes which are necessary for their own activity. Epigenetic changes are preserved when cells divide, and this is important to maintain the cell line for growth and repair. Now, epigenetic changes mostly occur within an individual organism's lifetime, but if gene inactivation occurs in a sperm or egg cell that results in fertilization, then some epigenetic changes can be inherited by the next generation. Now, due to the regulatory role during transcription of epigenetic modification in genes, it's not surprising that these changes in epigenetic markers, such as acetylation, can contribute to cancer development. So HDAC expression and activity in tumour cells is very different from that in normal cells. The overexpression and the increased activity of HDACs has been shown to be a characteristic of tumorigenesis and metastasis, in other words, the spreading of malignant tumours. And this suggests an important regulatory role of histone deacetylation on oncogene expression. Now, oncogenes are the genes which are implicated in the development of various types of cancer. Now, one of the examples of this is the regulation role of histone acetylation and deacetylation in the P300 and CBP genes, both of which contribute to oncogenesis. Now, in 2006, there was approval given by the US Food and Drug Administration to vorinostat, and this represents a new category for anti-cancer drugs which are in development. Now, vorinostat targets histone acetylation mechanisms, and this can effectively inhibit abnormal chromatin remodeling in cancerous cells. Now, this is sold and marketed under the trade name Zolinza for the treatment of cutaneous T-cell lymphoma, or CTCL, when the disease persists, gets worse, or comes back during or after treatment with other medicines. Now, hypermethylation or hypomethylation of DNA when compared to normal tissues has been linked to a large number of different types of human cancer. Now, it's known that the 5-methylcytosine present in the methylated CpG islands that we referred to before can itself be converted by deamination into thymine. Now, this is a potentially permanent mutation, and so it's effectively a substitution of the base sequence, and this could well lead to cancer. So, methylation of DNA or histones causes the nucleosome, as we said before, to pack more tightly together. This in turn prevents transcription factors from binding with the DNA, and this in turn prevents those genes from being transcribed and therefore expressed. So, histone acetylation will result in more loose packaging of adjacent nucleosomes, and this in turn allows transcription factors to bind with the DNA and therefore allow those genes to be expressed. Now, we've spoken quite a lot about the underlying regulatory mechanisms which can lead to cancer, but we haven't really talked very much about cancer as a condition. So let's start with an introduction to cancer. So a tumour, or a neoplasm, is tissue composed of cells which have grown and multiplied abnormally. Now, in normal tissues, mitosis is a tightly controlled process, and this leads to the regulation of the rate of the growth of those normal tissues. Now, when these regulatory processes that control the rate of cell division break down, the cells can then start to divide uncontrollably, and this is what leads to the formation of tumours. Now, tumours are, again, sometimes referred to as neoplasms. So tumours can be categorised as either benign or malignant. Now, it's actually the malignant tumours which are classified as cancers. Benign tumours can remain localised, they tend not to spread, and they tend to be very well demarcated. In other words, the edges of a benign tumour are very clearly visible in contrast to the surrounding healthy tissue. Now, as such, benign tumours are very rarely lethal, but they can be, because as we'll see, they can impinge on the or rather they can press on surrounding structures, and this in turn can lead to serious conditions, particularly inside the skull and other areas where the movement of the surrounding healthy tissue is restricted. So benign tumours tend to remain localised as discrete masses and are rarely fatal. Now, benign tumours, as we said before, they can grow and become larger, and in this sense, this is when benign tumours themselves can cause problems. Now, malignant tumours are cancers, or classified as cancers, and these display very different characteristics to benign tumours, most notably their ability to invade and spread to surrounding tissues. So, cells of benign tumours have specific abnormal characteristics other than just uncontrolled cell division. So these cells tend to be undifferentiated or unspecialized, and the cells tend to separate readily from the tissue and then spread via the blood or the lymphatic systems to other tissues where they can form secondary tumours. Now, it's this process of the spreading of a primary tumour via the blood or the lymph to other locations in other tissues of the body. This is referred to as metastasis. So cancer cells can cause considerable damage and can compete with the normal tissue for nutrients. So here is a map which shows the worldwide incidence of various forms of cancer worldwide, and I'll leave that up there for you guys to have a look at while I take another sip of my tea because I'm rapidly losing my voice. Okay. So let's have a look at some of the contributing factors which relate to the incidence of cancers. Now, the four most common cancers are breast cancer, lung cancer, colorectal cancer, and the cancer of the prostate. Now, in 2003, breast cancer accounted for about 32% of new cancer cases among women, and prostate cancer accounted for approximately 24% of cancers among men. Now, the susceptibility of individuals to cancer is a result of a combination of different factors. Now, the main factors that affect the risk of developing particular kinds of cancer are the genetic makeup of an individual, the environmental exposure to carcinogens, the personal lifestyle of the individual, the age and the gender of the individual. These are all the main factors which contribute to the risk of a particular individual developing a particular kind of cancer. Now again, a carcinogen is an agent which increases the likelihood of cancer. So carcinogens tend to be mutagens like radiation, chemicals, and viruses. So some of the factors that contribute to cancer are listed here, and none of these will come as a particular shock to any of you who've studied this topic previously. Now, before we go any further into the genetic mechanisms behind cancer, we need to be aware that there are different forms of cancer, and they are named in a particular way in medicine. So cancers, again, the term cancer refers specifically to malignant types of tumour. These are categorised in accordance with the functions and the locations of the cells from which they originate. So the term carcinoma refers to a tumour derived from epithelial cells. So these are the cells, again, that line the surfaces of various body organs. So for example, the skin, airways, digestive tract, and this is the commonest form of cancer, which accounts for between 80 and 90% of all reported cases. Now, the term sarcoma refers to tumours derived from bone, cartilage, muscle, fat, or connective tissues. Leukemia refers to cancers of white blood cells or their precursors, and again, the precursor cells are manufactured in the bone marrow. Lymphoma is a type of cancer where the cancer cells are derived from the bone marrow, and this affects specifically the lymphatic system. Myeloma is a cancer of the antibody secreting B lymphocytes. Now, we go into a lot more depth on the epidemiology, the etiology, and the pathology of various cancers in that course module, which relates to this subject on the Invisible College website. So those of you who have access to that course module, please do look into that because there's a lot more detail there. But for the sake of brevity, let's just move on now to the mechanisms of genetic mechanisms underlying cancer. And in order to do that, we need to explore proto-oncogenes, oncogenes, and suppressor genes. So what is the relationship between genes and cancer? Now, cancers or malignant tumours result typically when the cell cycle cannot be controlled in certain types of cell. Now, cell growth and division is controlled by proto-oncogenes and also tumour suppressor genes. Now, proto-oncogenes can be thought of as the accelerators, and tumour suppressor genes can be thought of as the brakes, which relates to the rate of the cell cycle. So in other words, proto-oncogenes are the genes responsible for accelerating the cell cycle and the rate of division, and in contrast, tumour suppressor genes are the genes which are responsible for slowing down that cell cycle and the rate of cell division. Now, mutations in either of these types of genes can obviously therefore lead to abnormal cell growth and division, which is basically the root cause of most forms of cancer. So essentially we can think of the genetic mechanisms underlying the incidence of cancer as either a faulty brake or a jammed accelerator. Now, in normal cells, the timing of cell division is under strict control, and it involves a network of stimulatory and inhibitory signals which work together in a balanced way. And again, it's the proto-oncogenes which code for protein products which generally will stimulate or promote or accelerate cell proliferation and division. Whereas in contrast, the tumour suppressor genes will code for protein products that generally will inhibit and constrain the rate of cell division. Now, proto-oncogene products therefore can be referred to as the accelerators of cell division, and tumour suppressor gene products are the brakes of cell division. Now, I'm using these terms kind of euphemistically. Please do not use these terms like accelerator and brake without qualification in your exam question answers. Because again, without qualification, your examiner may not know what you're talking about, or if they do, they may not give you credit for you knowing what you're talking about. We're simply using these terms as a kind of euphemism to explain how these things operate here. So, mutated proto-oncogenes or tumour suppressor genes can then cause a faulty brake or a faulty accelerator, and a combination of these mutations can be lethal. So let's start with proto-oncogenes. So, there are numerous, about 70 proto-oncogenes which have been identified, and all of these are involved in the transduction of signals, the transfer of information from the outside of a cell to the inside. Now, many of these proto-oncogenes, such as the RAS proto-oncogene, these are responsible for providing the positive signals that lead to or promote cell division. So these are a class of genes that can produce dominance mutations and become oncogenes. So the term proto, or the prefix proto, means before. So for example, a protostar is the mass of material which before fusion ignites, or rather just after fusion ignites, can then cause the formation of a star. A proto-oncogene is the gene which can undergo mutation to cause a cancer-causing gene. And again, oncology is the study of cancer, so an oncogene is a gene that can potentially cause cancer. So you see where these kind of prefixes and the etymology of these terms. So cancers usually result from the accumulation of many mutations to more than one gene. So some of these mutations can be dominant and others can be recessive. So cells continuously communicate with each other and with their environment in order to ensure their normal functioning. So cells will respond to molecular signals such as growth factors in their environment, and this involves elaborate signaling pathways. Signals on the cell surface are recognized, transduced, amplified and transmitted along a variety of different pathways and chemical cascades, and all of these will affect cell division. Now, the RAS proto-oncogene codes for a plasma membrane G protein with GTPase activity. Now, as we saw before in the previous webinar in this sequence, these G proteins can act as molecular switches in signaling pathways, and these signaling pathways, among other things, control the rate of cell growth and also differentiation. So mutations in the RAS genes can create oncogenes that code for altered RAS proteins or GTPases. Now, these mutant proteins in turn will affect the molecular switching mechanisms that control cell growth, cell division, and also programmed cell death or apoptosis. So disruption of a signaling pathway through the mutation of the RAS genes, is involved in many different cancer types, including about half of all colon cancers and about 90% of all pancreatic cancers. So this gives you a sense of just how important the disruption of these pathways is in the etiology and the incidence of cancer. So the RAS GTP protein is bound to GPP, and i.e. GDP in its off state when the signaling molecules are absent. So the signaling molecule or the growth factor will then bind to specific membrane receptors. The signal is then relayed from the receptor to the RAS GTPase protein. GPP is then expelled and exchanged for GTP. The GTP binding then changes the RAS protein and exposes the binding site for other molecules. So then a cascade of different biochemical events is triggered by this, and this in turn will then activate the processes involved in switching on cell division and cell growth. So, in the absence of the signaling molecule, the GTPase of the RAS protein will then catalyze the conversion of GTP back to GDP, and the molecular switch is therefore turned off. And in this sense, because of this, mitotic events are then halted, because when the RAS gene mutates to an oncogene, abnormal RAS proteins with defective GTPase activity are formed. The GTP fails to be hydrolysed back into GDP even when the signals are absent. So in this case, what's happened here is the on-off switch has failed. Now the consequence of this is that the RAS protein is continually switched on, and this allows the cell to grow and divide in an uncontrolled manner. And this ultimately is what leads to cancer. So the Mike proto-oncogene codes for a protein product that acts as a transcription factor and controls the expression of several genes involved in the cell cycle. Now again, as we saw before, that gene is located on chromosome 8, and a particular mutation of this gene involves the movement, the translocation of the the Mike proto-oncogene from chromosome 8 to chromosome 14, as we saw before. Now this results in an overexpression of the Mike gene, and therefore the subsequent overstimulation of the cell division. So the Mike gene is now acting as an oncogene, and as we saw before, this is implicated in the development of a number of cancers, and most notably Burkitt's lymphoma. So here is again what's happened. As we saw before, we get this translocation which leads to an overexpression of the Mike gene, and therefore the Mike gene has now become an oncogene, which leads to the overstimulation of the cell division, which in turn causes Burkitt's lymphoma. Okay everyone, it seems like we've come to the end of the second hour of this webinar, so we're going to move on to look at tumour suppressor genes next, but just like before, I'm going to give you guys a 5-minute break. I see that we have lots and lots of questions to answer in the Q&A box. Now, remember that all of you who are in attendance who booked tickets for this webinar, you have all received a pack of resources, including a workbook containing a lot of these worked examples, and many of you have uploaded those to your private coaching areas, and if you now check your private coaching areas, you'll find that I have marked those workbooks and given you some feedback on those. So please do check those. Don't forget to check those, because there's a lot of feedback on what I've given you back. So after the 5-minute break, we're going to get into tumour suppressor genes, and we'll talk about the mechanisms by which those work as well. So again, take a 5-minute break, and I will see you in 5 minutes time. So we were talking about proto-oncogenes. Let's move on to talking about tumour suppressor genes now. Okay, so tumour suppressors, in contrast to what we were talking about with proto-oncogenes, these are genes whose protein products act to control cell division by inhibiting or slowing down the rate of cell proliferation. So, in contrast to the proto-oncogenes, these are essentially the brakes for cell division. So tumour suppressor genes, these negatively control cell growth, and the mutations of these genes will lead to the removal of that constraint, and therefore can lead to cancer formation. Now, again, hypermethylation of the DNA is one of the major epigenetic modifications which represses transcription via the promoter regions of a tumor suppressor gene. Now, again, as we said before, hypermethylation usually occurs at the CpG islands in the promoter regions and results in gene inactivation. Now, this in turn results in unconstrained cell proliferation, and again, this is at the core of the causes for cancer. Now, in contrast, hypomethylation has been also implicated in the development of progression of cancer through mechanisms which are different or distinct from DNA methylation. But a discussion of those mechanisms is frankly beyond the scope of this webinar here. Okay. So one of the most important tumor suppressor genes is the p53 gene. Now this was discovered in 1979, and it's actually found to be associated with almost 50% of cancers. So whenever we get mutations of this p53 gene, we find that these typically are at the root of almost 50% of known cases of cancer. Now this is located on chromosome 17, and the protein product of this gene again acts as a transcription factor. Now remember, a transcription factor is a protein which binds to a promoter region adjacent to a gene where it regulates the transcription of that gene. Now in this case, the p53 protein product has been described as the guardian of the genome. Now essentially what it does is it senses the health of the cell and its DNA. Now I'm using teleological terms like sense to describe something that an inanimate object is doing, but you get the idea here. Now the protein product of p53 essentially is involved in the detection of cellular and DNA damage. Now when it does detect the damage, it aids the cell in essentially making a decision. The cell has to decide when it detects this kind of DNA damage whether or not the cell should be allowed to divide, or whether it's safer to just kill that cell by triggering programmed cell death, which is referred to as apoptosis. Now it triggers repair to the damage where it's appropriate, and it brings about the cell death when the damage is so bad that further cell division would propagate the genetic defects and expose that organism to the risk of the development of tumours. Now, the non-mutated p53 gene serves a protective function against tumour formation. So how does this work then? So the p53 protein is a very specific transcription factor, and it induces the expression of about 6 genes, including a gene called p21. Now, one of these gene products, the p21 gene product, binds to the cell cycle control proteins called cyclins and inhibits them. Now this in turn will arrest the cell cycle at the G1S boundary, and this prevents the entry of the cell into the S phase of the cell cycle. Now normal healthy cells have very low levels of the p53 protein product, and the cell division is not inhibited therefore. Now when the DNA is damaged, the level of p53 protein increases, and hence the level of the p21 protein also increases. So cell division at that point is stopped or arrested at the G1S boundary checkpoint until the damaged DNA is either repaired, and if that's not possible, then the cell is then triggered into a stage of apoptosis or programmed cell death. Now, following the repair, if a repair is possible, the p53 and the p21 protein levels will fall, and then the normal cycle is resumed. So, we can summarize this process by saying that when the DNA is damaged, the cell will respond by increasing the levels of the p53 protein which are expressed. This then arrests the cell cycle between the G1 and the S phase. This in turn can trigger DNA repair, or the damage to the region that was repaired, but if that's not possible, then the cell will simply respond by triggering the programmed cell death of that cell before that cell can start to divide uncontrollably and trigger the formation of a tumour. So this is actually an incredibly clever mechanism. Now I use the term clever in inverted air quotes just because I dislike the use of teleological language. In other words, I dislike the use of language by scientists which ascribes human characteristics to inanimate objects. Now we're talking about molecular mechanisms here, which are themselves unconscious because cells lack consciousness. But nonetheless, you see just how effective and complex these mechanisms can be. Cells are incredibly complicated machines, essentially, which will respond to changes in their external and internal environment, including repairing themselves or, if necessary, triggering their own death to protect the multicellular organism. So we see here that p53 plays a major role in preventing tumour development, and it's in this context that these types of genes are referred to as tumour suppressor genes. So mutation of the p53 gene is actually one of the most frequent genetic changes seen in cancer cells. Now, the effect of a mutation of the p53 gene depends on its location within the gene. The protein product p53 of the gene is then affected in different ways. Now, the overall effect of most p53 gene mutations is either a decrease or the absence of the p53 protein product in the cell. Now, the protein may be produced in an inactive form, or its rate of breakdown within the cell may be greater than normal under conditions of a gene mutation in the p53 gene. So mutant p53 proteins are generally unable to function as effective tumour suppressors. So we see here that we have transcription and translation of the p53 gene, which can, under normal circumstances, will lead to the active protein. But if you have a mutant p53 gene, this can lead to an inactive form of the p53 protein being translated. So two p53 proteins can combine to form a dimer. Now, the final functional p53 protein must contain two active protein products. So this would be the active p53 dimer, and these are all the different combinations of inactive p53 dimers, and these again will lead to potential tumour formation. So the effect of mutant p53 protein we can summarise as follows. You get damage to the DNA, the p53 levels fail to increase, and therefore the cell can replicate with the damaged DNA, and the cell cycle fails to be arrested, and the cells undergo uncontrolled division, and as more mutations accumulate, the cells can become cancerous. So a lack of p53 function in cells can result in a loss of the ability to eliminate potentially cancerous cells, and tumours can form as a result. So going back to the genetics of breast cancer, we can see here that most breast cancers are sporadic, with around 8% being familial or hereditary cancers. Now, the incidence of breast cancer varies with age, with the highest number of cases being diagnosed in the age group between 50 and 64 years of age. So, in terms of breast cancer, it's mutations in the BRCA1 and BRCA2 genes which are associated with some cases of familial or hereditary breast cancer. Now, when functioning normally, these genes will promote DNA repair mechanisms and therefore will arrest cell division under conditions where the cell is likely to become cancerous. Now, if one copy of these genes is mutated, the other copy can act as a brake on uncontrolled cell division. So, a spontaneous mutation that removes the only functioning copy of the genes increases the risk of tumour development. So in this context, again, BRCA1 and BRCA2 can be described as tumour suppressor genes. So these genes are described as breast cancer susceptibility genes, although mutations in these genes are rare, only accounting for a minority of the familial breast cancers. So ongoing repair of DNA will maintain normal cell function and will also avoid cancer. It's the failure of the repair system which can itself lead to cancer. So if we have DNA bases modified or damaged, so for example by the methylation of cytosine, this in turn can lead to the detection of the damaged base and the removal of that damaged base, which in turn removes neighbouring nucleotides, which in turn leads to the ultimate repair of that DNA. Now, it's actually estimated that many thousands of these kinds of repairs and modifications can occur in a healthy single cell every single day. So in other words, your cells are constantly being damaged in these ways, and yet the normal repair and maintenance mechanisms operating inside your cells are so effective that for the majority of us, none of us will go on to develop cancer as a result of these thousands of such damages and repairs taking place in each one of your healthy cells every day. Now, more recently, scientists have identified genes whose mutations are more significant than the ones that we've already mentioned in increasing the breast cancer level of risk. So attention has shifted to the so-called CHEK2 genes, which are isolated on chromosome 22 and mutations of that gene. So here we can see a molecular model of a mutant CHEK2 protein. So the CHEK2 gene produces a protein product which is involved in the regulation, again, of the cell cycle. Now, the CHEK2 protein is activated, again, when the DNA becomes damaged. So this protein interacts with the tumor suppressor protein p53, and it's this interaction which arrests the cell cycle in response to DNA damage. So the CHEK2 protein also regulates the action of the BRCA1 protein. So all these protein gene products play important roles rather, in determining whether a cell will repair the DNA or will undergo normal programmed cell death or apoptosis. So cells with mutated or damaged DNA are therefore prevented from dividing uncontrollably, and in turn this prevents the formation of tumours. So mutant CHEK2 protein removes that tight control of the cell cycle and therefore makes cancers and the development of tumours more likely. So the CHEK2 protein is actually an enzyme. It's a kinase which phosphorylates and therefore activates both BRCA1 and the p53 proteins. Therefore, the mutant CHEK2 protein fails to activate these tumour suppressor proteins, so cells with the mutant CHEK2 protein will therefore replicate with the damaged DNA as the cell cycle fails to be stopped. So we can summarise what we've been talking about by saying that when the DNA is damaged, the mutant CHEK2 protein is unable to activate the p53 and BRCA1 tumour suppressor genes. This in turn leads to the failure for these proteins to be activated, and these therefore fail to prevent the cell from entering uncontrolled division, and the cell therefore can then potentially divide uncontrollably and therefore form tumours and therefore cancer. So the next section of the spec that we need to deal with is the section which goes into genome projects. So let's move into that now. So we need to again contrast the genome with the epigenome. So to remind you, genetics is the study of biological processes and phenotypes and their functional molecules encoded as heritable gene sequence information in the genome. So the genome of a species refers to all the genes present in that species. So genomics is the study of an organism's entire genome. Now in contrast, epigenetics is the study of stable cellular and physiological traits and their underlying mechanisms that are heritable by daughter cells and which result from changes in gene expression which are not due to alterations in the DNA nucleotide sequence. So the sum of all these epigenetic changes in a cell is known as the epigenome. So here we can illustrate the number of base pairs in 4 very different genomes. So in a virus, we're looking at 5 times 10 to the 3rd base pairs. In a bacterium, we're looking at 4 times 10 to the 6th base pairs. In a Drosophila fruit fly, we're looking at 1.3 times 10 to the 8th base pairs. And in a human, it's 3 times 10 to the 9th base pairs. So the knowledge gained from genomics is a major resource to genetic engineers and also the pharmaceutical industry. Genomics is allowing us to produce new medical drugs and also to develop new methods of pest control, among other applications. Now, another term that you need to be familiar with is the term proteome. Now, the proteome is the entire set of proteins expressed by a genome, cell, tissue, or organism at a certain time under defined sets of conditions. So again, this goes back to the so-called central dogma of biology, whereby the information in DNA is copied to mRNA by the process of transcription. The information in mRNA is then translated into a functional protein by the process of translation. So the term is a portmanteau of proteins and genome. So proteomics is the study of the proteome. So the proteins are identified by two main processes. So the first of these processes that we're going to be making reference to in this presentation is gel electrophoresis, but there's also the process of mass spectrometry. Now we're not going to get into mass spectrometry because frankly that's way more chemistry than it is physics, but many of you will recall the process of mass spectrometry from your GCSE chemistry. Now, in May 2014, a draft map of the human proteome was published in the science journal Nature, and the map described proteins coded by 17,294 genes. Now, this accounts for approximately 84% of the total annotated protein coding genes currently known to exist in the human genome. Now, the human genome is currently believed to be composed of less than 20,000 genes that code for the diversity of proteins found within the species. Now, despite the large amount of DNA within the human genome, only 1.5% of that DNA actually codes for proteins. The remaining DNA consists of non-coding RNA genes, regulatory sequences, introns, and non-coding DNA. Now, non-coding DNA used to be called junk DNA, but now it's called non-coding DNA. So, in 1976, Walter Fiers of the University of Ghent in Belgium described the first complete nucleotide sequence. The sequence, that sequence was for the bacteriophage MS2, an RNA virus. In 1977, Fred Sanger of the Medical Research Council described the first complete DNA nucleotide sequence, and that sequence was for the bacteriophage X174 with only 5,386 base pairs. Now, the first complete genome sequences for representatives of all three domains of life were released during the mid-1990s. So, we have the phage virus here. Now, in 2003, Craig Venter was the first to completely assemble a fully functioning genome, that of this phage virus here, in vitro from synthesized oligonucleotides. So, again, this has a nucleotide sequence of 5,386 nucleotides, and these nucleotides code for only 11 proteins. So these kinds of advances in genomics have caused the modern preference for a three-domain classification system, and this led to a large reclassification, particularly within the kingdom Plantae. So this tree of life was based on DNA and RNA studies and was finally published in 2006. So the preferred three-domain classification system currently is represented here. We have the domain of the bacteria, the domain of the archaea. These are the two prokaryotic domains. And we also have the eukarya domain, which contains the familiar kingdoms that we referred to before. Now, all of these were thought to have been derived from a common universal ancestor. So determining the genome of pathogenic microbes gives us the knowledge of the DNA base sequence in a gene. We can then determine the amino acid sequence in its polypeptide or protein product, and that kind of knowledge can identify previously unknown antigens on the surfaces of pathogens, which in turn can allow us to develop things like new vaccines. So, the Human Genome Project was an international scientific research project which was proposed in 1984 by the US government, and to date, the Human Genome Project actually remains the world's largest collaborative biological project. Its goals were to firstly determine the sequence of the chemical base pairs that makes up the human DNA, and then to identify and map all of the genes of the human genome from a both physical and functional standpoint. Now, the project actually formally began in 1990, and most of the sequencing was done by the Celera Corporation and universities in countries like the United States, the UK, Japan, France, Germany, and China, and it was declared complete in 2003. However, by 2003, only 90% of all of the DNA found in human cells had been sequenced, and it wasn't in fact until May 2006 that the sequence of the last chromosome was published in the science journal Nature. So the knowledge gained from the Human Genome Project has been already the kind of fountainhead of major advances in medicine and biotechnology and various other disciplines. So there are now genetic tests that can show if a person has a predisposition to a variety of illnesses, and these include breast cancer, haemophilia, cystic fibrosis, liver disease, and various other conditions. Now also the causes of many cancers as well as diseases like Alzheimer's disease are being discovered, and this should eventually lead to a revolution in the treatments of these conditions. Now the science of epigenetics is now offering us a way to understand the mechanisms by which some foods are associated with reducing the risks of developing certain medical conditions. So for example, in early 2015, sorry, got an eyelash in my eye, that's helpful. So, for example, in early 2015, researchers found that the biologically active molecule resveratrol, which is an antioxidant found in things like red grapes, red wine, raspberries and blueberries, this could prevent age-related mental decline and coronary heart disease. And this was demonstrated fairly conclusively in rats, but of course rats are so biochemically and physiologically similar to humans that the conclusion could just as well be applied to humans, potentially. So, giving older rats suffering from age-related cognitive loss, this compound resveratrol clearly led to improvements in learning, memory and mood. So one of the characteristics of Alzheimer's disease is the development in the brain of amyloid plaques. Now amyloids can cause degeneration of nervous tissue and can consist of any one of 18 inappropriately folded versions of proteins and polypeptides which are naturally found in healthy individuals. So it's hoped that knowledge of the human genome and also relevant epigenetic data and other techniques will eventually enable us to identify the genes that control the production of these amyloids and also other unnatural proteins which can lead to pathologies. So a treatment to slow, to stop, or even to reverse the effects of diseases such as Alzheimer's disease or also Creutzfeldt-Jakob disease, so-called mad cow disease, eventually may be possible. So, where is all of this technology going then? Well, first of all, we now have mass public sequencing. This actually has immense implications because it eventually will enable us to radically improve our understanding of the underlying genetic causes of diseases. So the huge databases obtained from public sequencing will eventually provide us with data which can be statistically analysed. And these results could potentially enable us to correlate specific genes with specific genetic traits and diseases. However, this kind of potentially world-changing technology is not without its concerns. Now, knowledge of the human genome or a personal genome cannot easily be translated into the proteome because of the presence of non-coding DNA and regulatory genes. So it's hoped that with new technologies, data obtained from a patient's whole genome sequence will eventually reveal information about disease conditions that the patient may have or will develop in the future. Now obviously this has certain ethical issues associated with it, because essentially how do we deal with this information? Is the information confidential to the individual, or do closely genetically related family members, for example, have a right to be informed about these? But there are even wider issues here, because for example there are already companies like for example 23andMe where you can send a sample of your DNA and they will sequence that ostensibly for use in determining your family's genealogy. However, there are numerous court cases in the United States, for example, which show that when this type of information is submitted to these companies will retain intellectual property rights over that genetic sequence. So essentially, your genetic information becomes somebody else's property. So you might want to consider whether or not that's ethical, whether or not that's desirable, and also obviously this opens up the possibility that your genetic sequences can be bought and sold between other companies without your knowledge or your consent. Now, there is a lot more information on current technologies which are involved in gene sequencing. So competing, for example, competing automatic automated next generation sequencing platforms have been launched over the last few years by various companies. Now, it was only about 10 years ago that to purchase a sequencing machine that would allow you to fully genetically sequence somebody's sample of DNA may have cost you several £1,000,000. Now, the costs have come down considerably. The size of these machines has been reduced considerably. So these automated next generation sequencing platforms are developing very rapidly, and they're producing systems which are more efficient, more powerful, smaller, and more cost effective. So, for example, a widely used next generation sequencing technology comes from a company called Illumina, and it's called the bead array process. Now, this makes use of 3-micron silica beads in microwells on either fiber optic bundles or on planar silica slides. Now, in February of 2014, this company's HiSec X10 system was the first one to produce a $1,000 human genome. Now, to put that into context, just a few years prior to that, you needed to spend millions of dollars to sequence somebody's complete genome. But here in 2014 was a company that produced a machine that could sequence an entire human genome for under $1,000. So this was a milestone in achieving low-cost sequencing based on the detection of, or based on these technologies. Now, in fact, these technologies have been taken further since then. So, for example, ion semiconductor sequencing is another one of these next-generation sequencing technologies, and this one is based on the detection of hydrogen ions, which are released during the polymerization of DNA. Now, this is a method of sequencing by synthesis, during which a complementary strand of DNA is built based upon the sequence of a template strand. So ion semiconductor sequencing is exemplified, for example, by the ion torrent system, which is a sequencing system produced by a company called Life Technologies. Now, due to recent cost reductions, the whole genome sequencing has now become a realistic application in DNA diagnostics. So, for example, another company, Oxford Nanopore, have produced a mini ion sequencer, and this is actually the size of a USB memory stick. So this gives you an idea of just how quickly these DNA sequencing technologies have moved forwards. Now, I'm not going to go into the details of those technologies in this presentation. If you want more detail on that, please do go into that course module on my website where we go into that in a lot more depth. So let's move on now, and the next section of the specification that we need to go over here is the use of recombinant DNA technologies. So we'll begin by the use of genetic fingerprinting, or the use of DNA technologies in genetic fingerprinting. So recombinant DNA technology. This is all about the intraspecific and interspecific manipulation of genes. So this involves the isolation of DNA sequences from one organism and the combination of those sequences with the DNA from a different organism. So this allows organisms to express genes and therefore manufacture proteins that they would not normally produce. So their DNA has been engineered to produce a product which is foreign to their natural makeup. So this process of recombinant DNA technology involves introducing segments of DNA or genes into a host organism by means of a carrier or a vector system. So the foreign DNA or gene becomes a permanent feature of the host, such that during the replication of the host cell, that gene, that foreign gene, is also passed on to the daughter cells along with the rest of its own DNA. So the foreign gene is therefore being cloned by this process. Now, one of the applications of this is the manufacture of hormones like human insulin and also human growth hormone. So how is this done then? Let's have a look at the procedure for doing this. So the first thing that needs to happen is that we need to obtain the gene that codes for the desired protein product. The next thing that happens is we need to select a suitable carrier or vector for transferring that gene into the host cells. Now, the most commonly used vectors are either bacterial plasmids or viruses. Now, whichever vector you choose, the next phase obviously is to insert that gene into the chosen vector. The next phase would be to allow the vector to carry the genes into the host cells. Now again, commonly used host cells are things like bacteria and yeast cells, and the reason these are chosen is for their rapid rates of reproduction and to allow that gene to be cloned and amplified very rapidly. Now the next phase would be to locate the host cells that have successfully taken up the desired gene, and then to culture those transformed cells on a commercial scale, usually using large fermenters. Now, once you do that, the final stage is to isolate and purify the desired gene product, and this is referred to, or the next phase would be referred to as downstream processing. So this animation that follows, if I bring that up on the screen, kind of illustrates this process. So let me make that a little bit bigger so you guys can see it. So that's the first step. This would be the second step. This would be the third step. And then we add the vector, in this case a plasmid, into the host, in this case a bacterium. We then allow that bacterium to start producing insulin and also multiplying to produce more bacteria. And the bacterium reproduces, and therefore the amount of the product, in this case insulin, produced rapidly increases, and then we can simply extract and isolate the purified product for whatever use we choose to put that product to. So we need to talk about the genetic engineer's toolkit for doing all of these things. So the genetic engineer uses 5 basic tools during the procedure for recombinant DNA technology. Firstly, there are restriction enzymes, which are enzymes capable of cutting DNA at specific sequences. The next would be the isolated genes that code for the desired product, and obviously we need vectors to carry that desired gene into the host. And again, these can either be circular plasmids from bacteria, or we can also use viruses as vectors. We also need an enzyme which is capable of splicing and gluing that desired gene into the vector. And finally, we need a host that will take up the vector in order to contain the gene and reproduce rapidly in order to produce many copies of the gene, and typically bacteria such as E. coli can be used for this purpose, but also yeast as well. So let's look at the first of these tools, the restriction endonuclease. Now, restriction enzymes, or endonucleases, these are groups of enzymes found in bacteria which recognize specific DNA sequences of between 4 and 6 nucleotides and make incisions or cuts within those sequences. So the specific nucleotide sequence recognized by the restriction enzymes are called restriction sites, and usually these are in the form of palindromes. In other words, sequences of bases which can be read, which are read exactly the same from one end to the other or in the opposite direction. So this would be an example of one such sequence, and as you can see, whether you choose to read it in this direction or in that direction, the palindrome has the same sequence. So in this case, the restriction endonuclease would cut at this location. So an example of one of these is the restriction endonuclease EcoR1, and this makes a double-stranded cut between A and G nucleotides on either side of the central axis. So this is where the enzyme cuts between A and G, and because it's palindromic, it will cut on the same side, or rather at the same position on either side of the palindrome, and this produces the ends of the DNA like this. Now, these cuts are staggered and they produce what we call sticky ends, because these 4 nucleotides, for example, are still complementary and could still bind with these nucleotides, but they can equally bind to another complementary sequence, and this is going to be important, as we'll see, in the use of these restriction endonucleases in transgenic technologies. So these are referred to as sticky ends. Now, some restriction enzymes, such as HPAL or HPAL, these cut the DNA strands at positions directly opposite to one another, and these, in contrast to sticky ends, will give so-called blunt ends. So HPAL recognizes the nucleotide sequence GTT-AAC, and it cuts between the T and the A nucleotides, again around the central axis, but in this case it produces what we call blunt ends. So there are over 700 different restriction enzymes which have now been identified and isolated from bacterial cells, and each enzyme is named after the bacterial strain from which it's been derived. So EcoR1 is from Escherichia coli strain RY13, and BAMH1 restriction endonuclease comes from the Bacillus amyloliquefacins strain H. So restriction enzymes that generate sticky ends are very, very useful to the genetic engineer. Now, the same restriction enzyme recognizes the same nucleotide sequence in the DNA from different species and therefore will create the same sticky ends. Now, when the DNA fragments from the two different species are then mixed together, the complementary bases on their sticky ends will be attracted to one another, and because they're complementary, they can form hydrogen bonds. So in this way, we can join the DNA fragments from different sources and join them together. So obviously this is going to become important because we need to take the gene from one organism of one species and splice it, for example, into a plasmid from a bacterium. Now, DNA ligase, or ligase, depending on who you ask, is the enzyme that seals the fragments of DNA together. So here we have a fragment of DNA from species X produced by a particular restriction endonuclease. If we then hit the DNA from another species Y with the same restriction endonuclease, it's going to produce complementary sticky ends. So the complementary bases from the sticky ends of the DNA from the different species will attract each other when they're mixed together, and they will form hydrogen bonds here, but we still need to seal these gaps in the sugar phosphate backbone, and for this reason we need DNA ligase. So now we've formed recombinant DNA, that is, DNA that contains gene sequences from two different species of organisms. Now, the other part of the genetic engineer's toolkit, as it were, is the use of vectors and plasmids. So vectors are carrier DNA molecules into which the DNA fragments containing specific genes can be inserted. So vectors are the means by which selected genes are carried into host cells where the desired gene is then cloned. So the isolated plasmids from bacterial cells and also the DNA from bacteriophage viruses are frequently used as vectors. Now again, to remind you, plasmids are small, circular, self-replicating, double-stranded DNA molecules found in bacterial cells which are separate from the main bacterial chromosome. Not sure why that diagram is not showing up. There we go. So here is the main chromosome of a bacterium, and to remind you, the plasmids are these smaller, circular rings of DNA. And these circular rings of DNA, which we call plasmids, are normally used by bacteria to transfer genetic information between each other. So this electron micrograph shows a single bacterial plasmid extracted from E. coli. So genes coding for desirable products can be spliced into plasmids to form recombinant plasmids. So when these plasmids are taken up by bacterial host cells, they will replicate along with the host cell and then will therefore clone that desired gene. So the plasmids can be obtained from bacterial cultures by breaking open the bacterial cells and then separating out the plasmids by ultracentrifugation. So the homogenized bacterial cells, when subjected to centrifugation, can then provide the plasmids into which the foreign genes can be inserted by the genetic engineer. So we can take the total human DNA extracted from human cells, and again this would be referred to as the genomic DNA or the genome. And now both the plasmids and the human DNA are treated with the same restriction enzyme so that the DNA from both sources will have complementary sticky ends. The two DNA molecules are therefore attracted to one another in the presence of DNA ligase, will then form a recombinant plasmid. So when the host bacterial cells are mixed with these recombinant plasmids, they take them up and become transformed. So these bacterial cells are now described as transgenic organisms because they express the genetic material from a different species. So when the transformed bacterial cell is allowed to divide, the recombinant plasmids will also replicate, and you'll find them in the daughter cells. And of course bacteria will divide multiple times very rapidly, and so you are essentially amplifying that gene. So in this sense, the foreign DNA has now been cloned. Now, we mentioned that bacteriophage viruses, and bacteriophages, typically lambda phages, is another useful tool to genetic engineers for cloning genes. So you find the linear DNA molecule packaged into the head group of the bacteriophage. So the phage DNA can then similarly be digested with a restriction enzyme to remove the central portion. The foreign DNA containing the desired gene is then spliced into the viral DNA. The recombinant DNA molecule is then packaged into the head of this lambda phage. And then we can allow the lambda phage to inject its DNA into the bacterial cell. And then the foreign DNA will then be inserted into the main chromosome of the bacterial cell. So the foreign DNA containing the desired gene is then cloned when the bacteria divides. So there are many applications of these different kinds of genetic engineering technologies. One such application is farming, spelled with a PH. Now this is the process of using plant crops or livestock to produce biochemical products which are not normally made in those species. So one such example is the inclusion of a human gene that codes for an antibody into a sheep's genome so that the sheep will produce that antibody in its milk. And then, once you've produced enough milk, the antibody can be relatively easily extracted from that milk and used in medical therapeutics. So these stages describe the main stages in producing antibodies from sheep. So we start with the human gene that carries the instructions for making the antibody, and we identify the position of that in the human genome. We then select an appropriate restriction endonuclease to cut that gene out of the chromosome. So at this point, we can then insert that gene into the nucleus of a particular sheep cell. The nucleus can then be fused with an enucleated egg cell from another sheep. So we're taking this gene, inserting it into a sheep cell nucleus from one sheep, and then we take an egg cell or ovum from another sheep, remove its nucleus so that we can place that nucleus containing that foreign gene into that egg cell. We then fuse that nucleus with the egg cell, and this essentially forms a zygote. So we can then simply allow that zygote to develop by mitotic division to an embryo. But then we can implant that rapidly dividing embryo into the uterus of yet another sheep. So 148 days after that, a lamb is born, but this transgenic lamb now carries the gene in all of its cells. So when this lamb matures and produces milk, that milk will contain the human antibody. And by doing this with sufficient quantities of these sheep, we can generate large quantities of antibody that can be extracted and purified from that milk. So farming of transgenic cows has been used to produce milk which is low in cholesterol to produce a more healthy diet, for example. But there are many other applications of this technology in biotechnology. So for example, the cloning of plants and animals is generally viewed by gene technologists as a means to improve a particular breed or variety. Now, cloning is also seen as a means of introducing genes into animals so that they produce a wide variety of useful substances for human beings, as we've seen before. Now, in 1996, famously, the scientists at the Roslin Institute in Scotland created the much publicised Dolly the sheep. And Dolly was actually created by injecting a nucleus from a mammary cell of a sheep into an egg cell without a nucleus. Now, this fertilised egg developed into Dolly, who is therefore a genetic clone of the sheep from which that nucleus was taken. Now, the creation of Dolly aroused worldwide interest and also concern about the possibility of human cloning. So cloning and genetic modification of humans is banned by international law, and there remain ethical implications for the whole field of genetic technology. So some of these, for example, this shows some of the main concerns that people have about genetic technologies in general. So let's quickly have a look at each of these. So these are by no means exhaustive, but for example, genetically modified crops. Now the release of genetically modified organisms into the environment could disturb the balance of the ecosystem, threatening biodiversity. And of course, once you've released these organisms into the environment and they start to reproduce with non-transgenic organisms, those genes cannot be recalled and the damage to the environment is done and will spread. There's also the concern over genetically modified foods. Now, the consumption of genetically modified foods has been shown to have unforeseen effects in humans. And so, again, there are concerns over this, particularly because there are certain countries where the political lobbies of biotech companies, for example, Monsanto, have so much power over the political process that they've actually prevented effective GMO labeling on foods. And the biggest example of this is in the US, where despite many attempts by concerned citizens and activist groups to demand the labeling of foods containing GMO products, such labeling is still sadly absent from many processed foods. So if you're living in America and you're eating processed foods, the chances are that you are eating genetically modified organisms with all of the potential risks that that involves. Another concern is the concern over so-called designer babies. Now, the potential cloning of human embryos could be exploited by some to create designer babies where people specify in advance their preference for the characteristics of the babies that they have. And of course, there is also the concern over genetic warfare. Now, unfortunately, there are a couple of nations, I won't name which, you can go and look this up for yourself. There are a couple of nations already which are developing race-specific bioweapons. Now, that is terrifying. The idea of biological weapons themselves is already terrifying, but the idea that you could target those bioweapons to target people of particular ethnicities, for example, is even more terrifying. So there are a number of concerns. Now, the ethical and moral implications of biotechnology, you know, I mean, we could talk about this for several hours, but let's quickly have a look at some of the questions that ideally I'd like all of you to ask yourselves, because way more so than my generation, your generations are going to be subject to these technologies, whether you like it or not. So, for example, we could ask the question, what are the advantages and disadvantages of knowing in advance that you're likely to die of heart disease or cancer? Is it ethical to abort a baby which will develop an hereditary disease or a malformed body? In theory, one day it will be possible to improve the human race by aborting all embryos with undesirable genes and use genetic engineering to enhance things like intelligence and physique. What are the arguments for and against this possibility? Consider the idea that all research into genetic modification should be stopped because it is playing God or playing at God. Is it ethical to insert human genes into pigs so that we can kill them to obtain human hearts, human lungs, human livers, and other organs for transplanting into humans? Should the living organisms or the products resulting from gene technology become the commercial property through patenting? And in fact, biotech corporations in various countries have already actually succeeded in patenting living organisms. How should cultural, moral, and religious beliefs affect the use of genetic technology? Should they, in fact, affect that use? Should we engineer living organisms to create products useful to humans? Can we be sure that animals used for gene technology do not suffer? Is the cloning of human embryos for research purposes acceptable? Who will have access to the genetic information obtained about individuals, for example? And are all genetically modified foods clearly labelled to allow full and informed customer choice? Now, in the case of the United States, this is not the case. In other countries, it may or may not be the case, and I'll invite you guys to do your own research in that regard. So these are just some of the potential questions, ethical and moral quandaries. Now, none of these questions has a definitive answer, and everybody is going to come down on one side of the fence or the other with respect to any of these questions. But these are the sorts of questions that you need to be asking yourself, because again, the specifications of all of the major exam boards for this topic do specify that you need to be aware and be able to evaluate the potential moral and ethical questions surrounding some of these technologies, as you can see here. Okay everyone, it seems like we've come to the end of the third hour of this presentation. I'm going to give you guys yet another 5-minute break before I go into the Q&A. Alternatively, if those of you who are still in the private Zoom webinar room prefer, I will continue and we can cover the remaining topics in this webinar. So we've just covered this section. We now need to go into the use of DNA probes and genetic fingerprinting, and that will conclude this topic. So I will leave it up to you guys if you would prefer me to continue, or if you would prefer me to schedule an additional webinar for those of you in attendance via Zoom who've already booked tickets to cover the remainder of this topic, perhaps later today or tomorrow. It's entirely up to you. Or if you'd prefer me to upload an additional video to cover those topics to your private coaching areas. Now, I realize that some of you have emailed me recently and said that you feel that it's unfair for me to upload the part 2s of the previous webinars in this sequence to YouTube because you've essentially booked tickets, whereas the people viewing those webinars on YouTube haven't. I appreciate your concern, and if enough of you vote on that, then I will certainly not do that. But be aware also that those of you who have booked tickets, you have been able to interact with me, and you will obviously be you've been able to ask questions in the Q&A, which I've addressed off of the YouTube live streams. So those Q&A sessions following each of these webinars have not been available to the YouTube channel viewers. So again, if that is your concern and enough of you vote for me to not upload those part 2 videos to YouTube, I will certainly do that. But again, so I will give you guys another 5-minute break. If you'd like me to continue and finish off the rest of this topic, then I will do that. If not, then I will shut off the YouTube live stream, and as I've done before with the previous webinars, I will then address the questions that you guys have posed in the Q&A box here. Okay everyone, so let me put up the 5-minute break timer, and then we will come back and I will do whatever it is that you guys have expressed your preference for me to do. So let me bring up the 5-minute timer, and then we will either continue or not. So let's give you guys 5 minutes to make that decision, and I will see you in 5 minutes time. Okay everyone, we're back. So let me have a look at what everybody's saying in the Zoom webinar chat. Okay, so you would all prefer that I shoot an additional video to cover those 2 last topics and upload them to your private coaching areas on the invisible-college.com website. I'm happy to do that. It seems like many of you agree with the idea that I shouldn't upload the second halves of these webinars to YouTube just because you guys have booked tickets and paid for them. But again, please do consider the fact that because you did book tickets, not only were you able to ask questions, which I'm about to answer in the Q&A session now, but also you were provided with a rather extensive pack of revision resources for each of these webinars, containing, among other things, your workbooks with lots of exam questions, lots of worked examples, and also the ability for you to upload that work and have me mark it and feed back on it to you, which obviously the YouTube viewers do not have. So I see that the attrition rate has been relatively high for this webinar. There's only 18 of you left in the private Zoom room, but of those 18 of you, it seems like 16 of you agree with that idea. So of course I will honour that request, seeing as those 16 of you have also been attending all of the other revision webinars in this sequence. So I have uploaded some additional videos to the, excuse me, losing my voice, to the YouTube channel for the website. What I will do is I will not upload the part 2s of these videos to the YouTube channel. I will only upload those additional portions of these webinars to your private coaching areas on the Invisible College website. Sorry, YouTube channel viewers, but you know that's the breaks. We live in a democracy after all. Okay, well, so I'm going to stop the YouTube live stream now, and we're going to go into the Q&A session. So Those of you watching this as a recording via YouTube, thank you for watching. I hope this has been useful to you. Of course, you are quite welcome to ask any questions that you have on the YouTube channel at the appropriate webinar video page. And those of you who would like to access the resources for these webinars and also the course modules on my website in which these topics are taught in much more depth, please do consider visiting my website at invisible-college.com. It is a work in progress. I have only been working on the website for about a year, but there is already quite a lot of value there in those course modules. And of course, you can always contact me via email or register a free account on my website, where you can contact me via the internal social network on my website as well. So once again, thank you for attending, and I will see you in the next webinar.